Buongiorno a tutti, sono Fabio Naro, addetto scientifico dell'Ambasciata d'Italia a Brasilia. Benvenuti al seminario sulla collaborazione scientifica tra Italia e Brasile sul Covid-19. Ringrazio l'ambasciatore Francesco Azzarello, la dottoressa Camille Giarretta Sacchetti e tutti i colleghi che interverranno nella parte scientifica. La parte di apertura e di chiusura sarà possibile ascoltarla in traduzione italiano-portoghese e viceversa, mentre la parte scientifica sarà in inglese senza traduzione. Il seminario sarà trasmesso in diretta anche sul sito Facebook dell'ambasciata. Per motivi di tempo non abbiamo previsto la possibilità di porre domande agli intervenuti, ma se vi fossero commenti o domande particolarmente interessanti potete lasciarli nella chat. Purtroppo vi devo comunicare che il professor Luis Alcantara per motivi familiari non potrà partecipare a questo evento e gli facciamo i nostri migliori auguri perché li possa risolvere questi problemi. Quindi lascio la parola all'ambasciatore d'Italia a Brasilia Francesco Azzarello e vi rinnovo gli auguri di buon lavoro.
Grazie Fabio, buongiorno, buon dia a todos, faccio sempre echos. Confesso che aprire questo seminario su un argomento così delicato e di valenza mondiale mi dà una profonda emozione e sono sincero. Ringrazio di cuore le importanti autorità governative e scientifiche brasiliane, il Ministero della Salute, un particolare saluto alla dottoressa Camille, l'Università di Brasilia, la Fondazione Fiocruz ed italiane, il Ministero dell'Università e della Ricerca, il professor Gaudio, grande piacere, grande onore, l'Istituto Superiore di Sanità, il professor Bussaferro altrettanto, l'Istituto Nazionale per le Malattie Infettive Spallanzani, l'Università di Verona e l'Università di Newcastle in Australia, insieme a tutti gli scienziati coinvolti per la loro preziosissima presenza e contributo. Ringrazio inoltre tutti coloro che stanno assistendo, fra cui altri importanti scienziati, per aver voluto dedicare tempo prezioso al seminario. Tutti coloro che in genere non appaiono per il loro assolutamente fondamentale contributo organizzativo. Non è stato semplice organizzare questo seminario. E last but not least, come si dice in inglese, il consigliere scientifico dell'Ambasciata d'Italia a Brasilia, professor Fabio Naro, cui va il merito di aver avuto l'idea e la passione di organizzare il seminario. Aggiungerei con una battuta ironica l'incoscienza, perché poveraccio sono varie settimane che non dorme per via appunto dei pensieri e di tutto quello che è connesso. Grazie Fabio. Il Brasile è il primo paese per produzione scientifica nel continente latinoamericano, con punte di eccellenza mondiale. L'Italia e il Brasile hanno una solida tradizione di collaborazione scientifica in tanti campi, e nel corso della pandemia la cooperazione è stata vigorosa e di alto livello, ma di questo ci parleranno gli scienziati. L'obiettivo dell'ambasciata, con il supporto della rete consolare e del solido sistema Italia in Brasile, è di far conoscere il legame scientifico bilaterale con le sue differenze e somiglianze, rendendolo fruibile a chi fosse interessato. La scienza moderna e transnazionale e lo sforzo che si continua a fare dall'inizio della pandemia è impressionante. La scienza è fatta da persone, spesso giovanissime, ben rappresentate anche in questo seminario, con un dottorando ed una ricercatrice postdoc, che hanno bisogno di muoversi, parlarsi e formarsi laddove esistano le conoscenze. Siamo tutti partecipi delle esigenze spesso frustrate dagli eventi di studenti, dottorandi, ricercatori e professori che devono muoversi fra l'Italia ed il Brasile. Nonostante queste difficoltà, molto è stato fatto sul piano scientifico nella lotta al Covid e l'Italia segue con grande interesse l'apporto brasiliano ai grandi temi della ricerca mondiale sulla pandemia, quali ad esempio l'individuazione e lo studio delle varianti genomiche virali. In questi giorni ha destato molto interesse l'annuncio che l'Anvisa stia valutando l'approvazione della sperimentazione clinica di 2 vaccini brasiliani. Su questo punto è bene sapere che in Italia sono state sviluppate alcune piattaforme biotecnologiche utilizzate sia per la realizzazione di un ben conosciuto vaccino anti-Covid, già presente sul mercato mondiale, sia di un altro vaccino, attualmente in fase 3 di sperimentazione clinica. Nel prossimo futuro, la collaborazione dei 2 paesi in questo campo di ricerca applicata potrebbe essere strategica sia per il Covid-19 sia per altre malattie virali di comune interesse. Vorrei infine rammentare, qui consentitemi di essere per una volta partigiano, che l'Italia ha molto da offrire anche come partner industriale nel campo della farmaceutica e del biomedicale. L'Italia, forse non tutti sanno, è il paese leader, ripeto leader, in Europa nella produzione farmaceutica. Alcuni esempi: un'azienda italiana tra i leader mondiali nel campo degli emoderivati ha messo a punto insieme ad un'azienda israeliana un approccio terapeutico innovativo con immunoglobuline per i pazienti Covid che ha già completato la fase 1 e 2 della sperimentazione clinica. Nel biomedicale vantiamo il distretto biomedicale di Mirandola, considerato il più importante del settore Europa e terzo nel mondo, polo di imprese specializzato nella produzione di dispositivi medici quali apparecchiature e prodotti monouso per applicazioni terapeutiche. Aziende italiane sono all'avanguardia anche nel settore della refrigerazione a bassissima temperatura, così importante per la logistica di alcuni vaccini. Pochi esempi danno l'idea di quanto Italia e Brasile possano scambiare in tecnologia, opportunità e trasferimento tecnologico anche in questo difficilissimo. Cedo ora la parola al professore Naro con gli auguri più sinceri di buon lavoro e successo a tutti voi, a tutti noi. Grazie Fabio, grazie a tutti.
Ringrazio l'ambasciatore per il suo intervento e passo alla dottoressa Camille Jarretta Sacchetti, direttora di Scienza e Tecnologia del Ministero da Saúde brasiliano. A lei, dottoressa.
Bom dia a todos, buongiorno a tutti. Inicialmente, eu gostaria de parabenizar a Embaixada da Itália, na figura do embaixador Francesco Azzarello e ao professor Naro, pela organização desse simpósio virtual, cujo tema é de inquestionável relevância. Notadamente, este momento de crise sanitária global, inserir a ciência como base estruturante para o desenvolvimento em saúde configura-se como o único, mais racional e mais humanitário caminho. Com impactos globais sem precedentes na história, a pandemia de Covid-19 vem colocando à prova a nossa capacidade de resposta local, nacional e internacional. O sistema de saúde brasileiro, o SUS, Sistema Único de Saúde, é reconhecido como um dos maiores sistemas de saúde pública do mundo. E com a pandemia do novo coronavírus, foi possível constatar a força e importância do nosso sistema de saúde, que pelo fato de ser integral e universal, atende cerca de 70% da população, quase 148 milhões de brasileiros. Sob a gestão tripartite dos 3 entes, governo federal, estados e municípios, vem sendo possível fornecer assistência aos pacientes infectados pela Covid. Além disso, o campo de atuação do SUS, o Sistema Único de Saúde, também inclui o apoio ao desenvolvimento científico e tecnológico. Desde o início da pandemia, este Ministério da Saúde mobilizou rapidamente a comunidade científica nacional para ajudar a responder às inúmeras lacunas de conhecimento relacionadas à Covid. Definimos prioridades de pesquisa, articulamos redes de pesquisadores e apoiamos projetos de colaboração nacional e global, a fim de maximizar os resultados e obter consenso científico mais rapidamente. No Brasil, nós temos uma rede de pesquisadores bastante qualificados e instituições de ciência e tecnologia sólidas, como a Fiocruz e o Butantan, que além da produção de conhecimento, foram capazes de estabelecer parcerias com laboratórios internacionais para produzir vacinas contra o novo coronavírus no território nacional. A ciência precisou se adaptar. Os cientistas trabalharam em tempo recorde e hoje temos inúmeras vacinas sendo utilizadas ao redor do mundo. Mas os inúmeros desafios científicos permanecem. Estamos preocupados com as novas variantes, em especial a P1, já que precisamos entender desde questões básicas sobre, por exemplo, se essa variante altera a história natural da doença da Covid e até mesmo responder qual será a eficácia das vacinas frente a essas novas variantes. Considerando que o coronavírus não conhece fronteiras e que as lacunas de evidências globais precisam ser devidamente resolvidas, torna-se ainda mais evidente a importância da colaboração científica. Nesse sentido, a vantagem de redes internacionais de colaboração traduz-se na capacidade de coordenar estudos multicêntricos com procedimentos padronizados, os quais conferem maior credibilidade aos resultados dos estudos. O Brasil, por meio da sua rede de pesquisadores, está disposto a ampliar a colaboração com os cientistas italianos pela sua capacidade e competência. O Departamento de Ciência e Tecnologia, da qual eu faço parte, que integra a Secretaria de Ciência, Tecnologia, Inovação e Insumos Estratégicos do Ministério da Saúde, é responsável por coordenar ações de pesquisa, seja por meio do financiamento direto a pesquisadores, seja sintetizando evidências para subsidiar a tomada de decisão no âmbito do SUS. Para além da Covid, nós estamos interessados em cooperar em outras áreas de extrema importância, como a área da genômica e da saúde de precisão. Em outubro de 2020, o presidente da República lançou o programa conhecido como Genomas Brasil, que é um programa de ciência, tecnologia e inovação que objetiva preparar o país para a era de medicina de precisão e personalizada. O programa está estruturado em torno de 3 pilares finalísticos, que têm por objetivos promover o fortalecimento da ciência e tecnologia, da indústria genômica nacional e do sistema de saúde. Uma das metas é que, ao final dos próximos 4 anos, o país tenha um banco de dados com mais de 100 mil genomas completos da nossa população, que é bastante heterogênea. A constituição desse banco permitirá que cientistas possam fazer avanços importantes sobre o conhecimento do genoma do brasileiro e seus impactos para a saúde. E, nesse sentido, estamos dispostos a colaborar com diversos países, entre eles a Itália. Atuar no desafio relacionado à saúde, em especial ao enfrentamento à Covid, continuará a exigir cooperação em um âmbito global sem precedentes. A colaboração internacional parece ser a melhor ferramenta para conter a disseminação do coronavírus. E nesse sentido, Dr. Fábio, eventos como hoje são de extrema relevância para que o compartilhamento de informações científicas aumente a resposta à pandemia. Eu desejo que essa discussão seja proveitosa e que a cooperação científica entre Brasil e Itália seja fortalecida. Agradeço pelo convite em nome do Ministério da Saúde.
Ringrazio la dottoressa Camille Giaretta Sacchetti per il suo intervento e passo la parola al professor Eugenio Gaudio del Ministero dell'Università e della Ricerca, consigliere della ministra Messa.
Grazie, professor Naro, signor ambasciatore Francesca Zarello, autorità brasiliane ed italiane, illustri relatori, signore e signori. È un piacere e un onore per me essere presente a questo seminario per videoconferenza sulla collaborazione scientifica fra Italia e Brasile sul Covid-19 e poter portare quindi i saluti della Ministra dell'Università e della Ricerca, professoressa Cristina Messa, e di miei personali come consigliere della Ministra. I rapporti fra Italia e Brasile sono antichi e di grande non solo collaborazione, ma anche sintonia ed empatia culturale. Cultura comune che trae origine dall'origine latina della cultura brasiliana, che si è mirabilmente fusa con la cultura locale e che costituisce un legame profondo che ci consente di capirci e di sentirci a proprio agio. Gli italiani che sono in Brasile sono sempre stati accolti molto bene, si trovano molto bene e si sentono a casa loro. In particolare, negli anni si sono instaurati importanti rapporti di collaborazione fra le università italiane e quelle brasiliane. Da un punto di vista numerico, questi rapporti fanno sì che dopo Francia, Spagna e Stati Uniti, il Brasile sia il quarto partner scientifico ed accademico dell'Italia, con ben 962 accordi fra 72 università italiane e 198 università brasiliane. Prima di altri paesi come la Cina, la Germania, la Russia e la stessa Argentina. Anche la recente pandemia ha trovato Italia e Brasile in prima linea. Da un lato, purtroppo, come paesi fra i più colpiti. Virulenza e mortalità del SARS-CoV-2 hanno inciso pesantemente sulle nostre popolazioni. A fronte di questo c'è stato un grande impegno scientifico degli scienziati italiani e brasiliani, anche in collaborazione fra di loro, che ha sostanzialmente portato l'Italia ad essere la terza per produzione scientifica a livello mondiale sul Covid-19 e il Brasile al 19° posto, con risultati di grande impatto sulla conoscenza della malattia e del virus ed un significativo impatto anche nel miglioramento dell'approccio diagnostico e terapeutico. Ancora più in particolare, ad oggi abbiamo più di 350 lavori scientifici in comune fra ricercatori italiani e brasiliani sul tema, alcuni dei quali sono diventati punti di riferimento per lo sviluppo della ricerca sul tema specifico. L'Italia poi è stata in prima linea su una serie di tematiche che vanno dalla sanità pubblica a quelle sui fattori di rischio, dai modelli per la comprensione della diffusione epidemica del virus, dalla risposta immune alle basi scientifiche per lo sviluppo dei vaccini, dalla genomica virale ai test diagnostici, dallo studio delle proteine strutturali del virus ai meccanismi di ingresso cellulare. Tutta questa attività ha portato l'Italia ad essere la terza al mondo per produzione dopo la Cina e gli Stati Uniti. Questo impegno, che insieme al Brasile è stato solidale e convergente, è stato uno degli elementi di risposta più significativi della comunità scientifica alla sfida di una così violenta ed inattesa pandemia, anche se in realtà gli scienziati da tempo avevano previsto la possibilità che una pandemia si sviluppasse all'interno del nostro pianeta. Molti giovani ricercatori italiani si sono impegnati assieme ai colleghi italiani e viceversa. L'occasione della lotta alla tragedia della pandemia da SARS-CoV-2 è stata colta con spirito proattivo e propulsivo da parte dei nostri ricercatori, che ha portato a un ulteriore irrobustimento delle relazioni di collaborazione scientifica fra Italia e Brasile, che dovrà ulteriormente crescere negli anni a venire. L'auspicio è quindi che, una volta superata, come ci auguriamo al più presto, la drammatica situazione pandemica, con tutto quello che con essa comporta anche a livello di scambi internazionali, i ricercatori italiani e brasiliani, gli accademici brasiliani ed italiani possano continuare ad incrementare i loro rapporti di collaborazione e che i governi possano reperire ulteriori risorse per implementare gli scambi culturali, accademici e di ricerca fra i nostri paesi. Ci auguriamo anche che i nostri giovani studenti universitari possano aumentare le proprie occasioni di scambio culturale con un paese come il Brasile, ricco di cultura, di natura e di prospettive di crescita, sulla base della comunità di intenti e delle analogie antropologico-culturali che legano i nostri due paesi. Ed è con questo auspicio che ringrazio e saluto ancora Sua Eccellenza l'ambasciatore d'Italia in Brasile, tutte le autorità scientifiche, governative brasiliane ed italiane qui presenti, trasmettendo l'impegno da parte del Ministero dell'Università e della Ricerca ad una sempre maggiore attenzione e supporto agli scambi e alle collaborazioni scientifiche fra mondo accademico e della ricerca di Brasile ed Italia. Grazie a tutti, grazie per aver organizzato questa bella e interessante conferenza e buon lavoro. Grazie.
Ringrazio il professor Eugenio Gaudio e passo la parola al professor Brusaferro, presidente dell'Istituto Superiore di Sanità. A lei, professore.
Grazie, dottor Naro. Professor Naro, prima di tutto porgo un caloroso saluto alle autorità collegate con noi, ai colleghi e a tutti i partecipanti a questo convegno. In particolare un saluto a Sua Eccellenza l'ambasciatore Francesco Azzarello, alla dottoressa Camilla Giarretta Sacchetti, che dirige l'Ufficio di Scienza e Tecnologia del Ministero della Salute brasiliano, al collega e amico professor Gaudio, che rappresenta il Ministero dell'Università e della Ricerca, al console generale di Rio de Janeiro, dottor Paolo Miraglia, e al professor Naro, attaché scientifico. Per me è davvero un piacere e un onore poter essere qui con voi oggi a rappresentare l'istituto che ho l'onore di presiedere e sapendo quanto Brasile e Italia hanno in comune. Ritengo che questo convegno, e sono veramente grato agli organizzatori per l'opportunità di poter condividere queste esperienze, possa essere una grande opportunità per confrontare e approfondire l'impatto che la pandemia sta avendo sui sistemi sanitari dei nostri due paesi, a oltre un anno dal suo esordio, ma anche è un'opportunità straordinaria per iniziare ad esplorare le opportunità di ripresa che si prefigurano nel post-Covid, alla luce delle strategie vaccinali che abbiamo posto in essere in Brasile ed in Italia. La pandemia di SARS-CoV-2 sta rappresentando una sfida epocale per i nostri sistemi sanitari e per i sistemi sanitari di tutto il mondo. E le implicazioni e le conseguenze vanno studiate e vanno, credo, è importante anche poterle in qualche modo prevedere. Non occorre che sottolinei quanto significativo sia stato l'impatto per l'Italia, primo paese occidentale a dover fronteggiare questa pandemia globale. Tuttavia voglio sottolineare come il Servizio Sanitario Nazionale del nostro paese, di cui andiamo molto orgogliosi, in tutte le sue componenti, a partire dai professionisti della salute, grazie all'impegno e alla sinergia tra istituzioni nazionali e regionali, ha messo in campo una rapida e coerente risposta istituzionale, che si è declinata attraverso l'adozione di strumenti e provvedimenti straordinari, diciamo pure unici nella storia recente del nostro Paese. Mi piace anche sottolineare il grande sforzo dei nostri concittadini nell'aderire a delle misure preventive di controllo proposte dal governo, e mi riferisco in particolare a quelle più restrittive, i famosi lockdown, che soprattutto nei primi mesi della pandemia erano momenti di veramente grande restrizione delle libertà personali. Il Servizio Sanitario Nazionale si è rapidamente adattato per non rispondere a questa sfida, in tutte le sue articolazioni. Il livello ospedaliero, nell'assistenza territoriale domiciliare, nei dipartimenti di prevenzione, che nel nostro sistema sono deputati all'attività di prevenzione e controllo della diffusione dell'epidemia, ivi comprese la grande attuale sfida della vaccinazione di massa. Come sapete, in Italia portiamo storicamente in doto la cultura del lazzaretto e della quarantena, che 600 anni fa la Repubblica di Venezia aveva messo in atto con successo, per diventare poi il primo Stato europeo a evitare la diffusione, in quel caso, della peste. Ma dobbiamo soprattutto ricordare che leggendo quanto hanno costruito e studiando quanto hanno realizzato i nostri avi, ancora oggi molte delle misure da loro messe in atto sono ancora efficaci nel contrasto anche di questa pandemia. La dottoressa Riccardo, nel corso del convegno, che ringrazio per la disponibilità a partecipare, vi rappresenterà l'aggiornamento sullo stato di diffusione dell'epidemia in Italia. Dai dati capirete come è ancor più necessario in questa fase della pandemia continuare a mantenere elevata la resilienza dei servizi sanitari, continuare a rafforzare la consapevolezza e la compliance della popolazione alle misure di contrasto al virus, realizzare una ricerca attiva e l'accertamento diagnostico dei potenziali casi di varianti, il sequenziamento cosiddetto del virus, ma allo stesso tempo percorrere programmi e perseguire con continuità e con urgenza per programmi vaccinali che riescano a intercettare il numero più elevato possibile di italiani nel più breve tempo possibile. In questo senso le attività di ricerca che offre questa pandemia sono notevolissime. Le quantità di domande a cui ancora dobbiamo rispondere sono ancora notevolissime e c'è veramente uno spazio molto ampio di collaborazione tra i nostri due paesi e tra i nostri ricercatori in questo settore specifico. Eventi straordinari come quelli che stiamo vivendo mettono in luce tutti gli aspetti più fragili dei sistemi sociali e sanitari, ma anche gli aspetti di resilienza e i punti di forza. E quello che impariamo da questa epidemia è che avere un sistema sanitario forte è fondamentale non solo per la risposta in questo caso alla pandemia da SARS-CoV-2, ma anche per lo sviluppo economico e sociale del Paese. Così come è importante e strategico avere forti centri di ricerca nel settore biomedico, avere una rete diffusa di laboratori per la microbiologia, per il sequenziamento dei virus, avere capacità nel produrre farmaci e vaccini, avere sistemi di allerta sulle emergenze sanitarie internazionali con capacità di raccolta e di scambio di dati epidemiologici. Proprio nella centralità della prospettiva internazionale, questa pandemia rende evidente ogni giorno l'opportunità e la necessità di approfondire la collaborazione e certamente anche la collaborazione tra i nostri due paesi può trovare forza e slancio in diversi settori proprio a partire dalla risposta a SARS-CoV-2. Penso tra l'altro al tema della formazione dei professionisti della sanità pubblica, ai diversi filoni di ricerca che vengono interrogati, vengono stimolati dalle domande ancora aperte che questa pandemia ci lascia e che progressivamente ci aiuta a formulare. Alla declinazione per esempio dell'approccio One Health nei programmi di sviluppo della sanità pubblica, così come agli obiettivi dell'Agenda 2030. Credo sia importante sottolineare la necessità di un grande investimento culturale e strutturale nei sistemi sanitari, a partire da un investimento sui professionisti, e ribadisco, credo che le persone in questo caso, oltre alle tecnologie, ma le persone fanno veramente la differenza, dei professionisti della sanità pubblica, sfruttando anche le grandi potenzialità che grazie purtroppo a questa epidemia si sono rivelate essere quelle legate alla formazione a distanza. La pandemia ci obbliga a favorire insomma e a progettare da subito il futuro dei nostri sistemi sanitari, quello italiano, quello brasiliano, un po' anche quelli internazionali, partendo dalla preparedness e dalla prevenzione delle attuali pandemie o delle attuali patologie prevalenti, ma anche tenendo conto che dovremmo far fronte a tutta una serie di patologie ben rappresentate dalle proiezioni al 2030 nel Burden of Disease, che una volta superata, speriamo rapidamente, questa pandemia, torneranno a diventare assolutamente prioritarie nell'agenda dei nostri servizi sanitari. Tutti questi temi poi vengono trattati e verranno affrontati nel Global Health Summit, che il 21 maggio vedrà riuniti virtualmente da Roma i capi di Stato e i ministri della sanità di tutto il mondo, sotto l'egida della Presidenza italiana del G20 e della Presidente della Commissione europea. Concludo con l'augurio di poterci ritrovare a ragionare di persona di tutte queste tematiche che il convegno oggi discute e che affronta in modalità virtuale. Concludo ancora con una nuova nota personale. Lo scorso dicembre sarei dovuto essere a Rio de Janeiro per la conferenza mondiale annuale degli Istituti Nazionali di Sanità Pubblica. Purtroppo l'epidemia ha stravolto tutti i nostri programmi. Ma con questa conclusione vuole essere anche un auspicio perché ci possano essere al più presto occasioni per incontrarci nei nostri due paesi. Vi auguro un buon proseguimento dei lavori e vi ringrazio ancora per aver organizzato questo evento. L'Istituto Superiore di Sanità, per il tramite del suo Presidente e attraverso tutti i suoi ricercatori, è ben felice e sarà ben felice di continuare a collaborare, ma anche di avviare nuove collaborazioni nel campo della sanità pubblica.
Ringrazio il professor Brusaferro e tutti gli intervenuti in questa prima parte del nostro convegno, del nostro seminario, per i loro interventi. We can move on to the scientific part, and I'm glad to introduce you the Professor Enrique Roberto Argañaras, professor di biologia molecular, coordenador do Laboratório de Neurovirologia Molecular da Faculdade de Ciências da Saúde da Universidade de Brasília, who will give us an insight about the molecular mechanism of COVID-19, talking about alpha-1 antitrypsin as a possible cellular factor against COVID-19. Please, Professor Argañaras, I give you the floor.
Thank you, Fabio. I share the screen and just it's okay.
Yes, it's okay.
Okay. Thank you for inviting me, and it's a pleasure to be here to talk about one of the projects that we have in collaboration with Professor Cipeto from Verona University. This project is concerning to the role of alpha-1 antitrypsin in the COVID-19 and its possible use for treatment of SARS-CoV infection. And I start with the current scenario of COVID pandemic, and until now, there are about more than 100 million infected people and more than 2 million deaths around the world as consequence of SARS infection. Among the main obstacles to overcome this challenge are the limited hospital capacity, the emergence of new viral variants, and the lack of effective treatment in addition to vaccine, and the lack of knowledge about molecular bases underlying the pathophysiology of COVID-19. Now I talk about a little bit about how cure the SARS-CoV infection. The SARS-CoV cell infection occurs in two steps, basically. The first one involves the binding of spike viral protein to the ACE2 receptor, and the second step involves the cleavage of spike protein by the TMPRSS2 protease and fusion with the host cell membrane. Regarding to the clinical manifestation and the early symptoms are fevers, headaches, dry cough, low smell, and diarrhea in some cases. That could be followed by the acute respiratory distress syndrome, characterized by pulmonary infiltrate, reduction of oxygen pressure, and acute dyspnea, and finally heart failure. Regarding to the pathophysiology of COVID-19, the COVID-19 is a multifactorial disease involving some comorbidities like cardiovascular disease, hypertension, diabetes, and chronic lung disease, as well as sex and age and genetic susceptibility. On the other hand, two molecular mechanisms are responsible for the pathophysiology of COVID. The first one is the acute inflammation known as cytokine storm, that is followed by disseminated intravascular coagulation. And there are basically three molecular mechanisms that are involved in the acute inflammation and disseminated intravascular coagulation. The first one is related to the viral infection itself. which induce the release in inflammatory cytokines, which in turn lead to damage and activation of endothelial cells, and which ultimately lead to activation of the extrinsic pathway and coagulation and thromboinflammation. The second mechanism involved the AC2 downmodulation as consequence of viral infection and by ADN17 overactivation, which lead to imbalance of renin-angiotensin system, that thus induce vasoconstriction and inflammation. The last one involved the pro-inflammatory cytokine release like TNF-alpha and IL-6 receptor, which lead to macrophage activation and release of other inflammatory cytokines, such as IL-1 beta and IL-8. And this is an overview that the pathophysiology mechanism involved in the COVID-19. The first one is the cell injury, the second is AC2 downmodulation, and the third is the ADN17 overactivation. Now I talk about a little bit alpha-1 antitrypsin, A1IT. Alpha-1 antitrypsin is a serine protease inhibitor, is a serpin family member, and expressed by hepatocytes, neutrophils, macrophage, endothelial pulmonary alveolar cells, intestine, and corneal cells. And the alpha-1 antitrypsin deficiency, there are more than 100 variants associated with different plasma level, and S and Z allele are more common and associated with a lower serum level. And interestingly, the middle-aged men show a decrease in alpha-1 antitrypsin plasma level, different than from women who present higher level, presenting significant variation during the age. Cigarette and pollution lead A1IT dysfunction too. And in 1 in 25 European descendants present Z allele, and 1 in 2,000 are homozygous for Z allele. Interesting, in northern Italy, in the Lombardy region, a high incidence of genotype C and S was observed. We must remember that this region had high mortality rate. Alpha-1 antitrypsin is a multitask protein. Maybe the main function of this protein is the anti-inflammatory protein. Inactivate the proteolytic enzyme neutrophils elastase, limiting acute lung injury. By instance, alpha-1 antitrypsin deficient are associated with chronic obstructive pulmonary disease. The other function is inhibit the binding of IL-8 to its receptor. The other function is inhibit ADN17 protease by inhibiting the release of TNF-alpha and IL-6 receptor. And the other important function is to inhibit the NF-kappa beta activation, that is a pro-inflammatory transcription factor. And the last and the important function too is the alpha-1 antitrypsin inhibit thrombin, that is serine protease and venous thromboembolism. And the other function, important function of this alpha-1 antitrypsin is the viral function. The alpha-1 antitrypsin inhibit HIV replication, and some genotype were linked with the hepatitis B and hepatitis C and HIV infection. And so what happened with alpha-1 antitrypsin in COVID-19? Alpha-1 antitrypsin inhibits SARS-CoV infection entry because inhibit the TMPRSS2, and this is the main function in the SARS-CoV infection. And the other function is inhibit ADN17 overactivation, mediated inflammation. So inhibit the release of cytokine, pro-inflammatory cytokine, like TNF-alpha and IL-6 receptor. and another important function is inhibit the AC2 down modulation. So this is an overview that the A1AT function COVID. The first one is inhibit the infection, as I mentioned before, and the other function is inhibit the ADN17 overactivation and the release of TNF-alpha and the pro-inflammatory cytokines. And another function is inhibit the thrombin, this important protein, serine proteinase in the coagulation. So in this context, our project, we have the following objective. The first one is determine the level of alpha-1 antitrypsin as well as ILC and TNF-alpha in the COVID patient with different clinical outcome. And the other objective is try to determine the prevalence of the alpha-1 antitrypsin deficiency variants like S and Z and their prevalence in the patient with COVID-19. And the last one, and I think it's more important, is that they develop the formulation like as nanoparticles for intranasal spraying and perform preclinical tests in COVID patients. So I think this is the brief of our project, one of our project, because we have several projects in collaboration with Professor Cipeto. And so thank you very much. Obrigado and grazie, I think. Okay, thank you.
Thank you very much, Professor Enrique Argañares, for your talk. It was very interesting. I'm sorry, but before, in another life, I was a professor. I would like very much to know about if you knew anything about the expression of alpha-1 antitrypsin genotype expression in some parts of Brazil, like Manaus, where we have seen such deep hit of a pandemic, but we will discuss it together, because it would be very interesting to make this correlation between Lombardia and Amazonia to see if there is something in common. Sorry, but I was a scientist. Okay, I go back to my...
It's interesting that, Fabio, that there are another study in silico that have been shown that the deficit of alpha-1 antitrypsin is overlapping with the region that was present more incidence of death by SARS-CoV infection. So there is a strong evidence. There are strong evidence that A1AT is important for in the SARS-CoV infection. Another important thing, this is protease inhibitor is the main local of action is in the lung. So I don't know, but I'm very confident that this protein is important, and we can use this protein in order to avoid the complication of SARS-CoV infection. You know?
Thank you very much. And I'm sorry to. and now I give the floor to Dr. Marta Giovannetti, who is a borsista di produttividade em pesquisa do SNPq no Laboratório de Referência Regional de Flavivírus do Instituto Oswaldo Cruz, Fundação Oswaldo Cruz di Rio de Janeiro. Dr. Giovannetti will talk about the crisis at the time of coronavirus, Italo-Brazilian cooperation to track emerging SARS-CoV-2 variants. And I asked Dr. Marta Giovannetti to give our best regards to Professor Alcantara. Okay, go ahead, Marta.
Okay, all right. So thank you, Fabio. Thank you so much. I will share just my screen. Okay. Can you see my presentation?
Yes, we can see it. Thank you, Mark.
Thank you. Thanks so much, Fabio. So first of all, I really would like to say thanks so much for the nice opportunity. So, and then of course, to try to bring all of us together. So actually the main objective of my talk is try to share our Italo-Brazilian cooperation that have been used to try to track the emerging SARS-CoV-2 variants in nearly real time. So in this sense, before starting, I really would like to say that we need to agree that for the first time during the COVID-19 pandemic, we have a nice example of international cooperation, right? So all of these make possible the availability and the generation of a huge amount of genomic data in just 13 months of epidemic, right? So we actually now have more than 920,000 complete genome sequence available of the SARS-CoV-2 that really give us the possibility to try to understand for the first time the connectivity of the country and how the virus was able to spread worldwide. So this is why I used to say that during the SARS-CoV-2 we really have the opportunity to perform for the first time this new phenomenal way to do science. Right? So everything was sharing lying in real time with the international research community. We know that actually since the first reporting cases, right, that have been detected in Wuhan in the Hubei province in China, actually we saw a really huge amount of SARS-CoV-2 cases that start to be notified worldwide, and all of these obviously allow the state of international concern and the state of pandemic by 11 March 2020. So this is actually the situation that we can see during this day regarding the total number of global deaths and the total number of total cases. So in this sense, I want to share, well, what about our contribution to the pandemic? So actually we were working since the first months of the epidemic, at the beginning and then pandemic, of course, between Brazil, the Campus Biomedical University of Rome, as well as the CRISP Institute in South Africa. So actually, I need to say that I was a bit lucky to have the possibility really to share myself in these three different sites, right? Well, in Brazil, I used to work in the Fiocruz, in the Oswaldo Cruz Foundation, which is located in Rio de Janeiro, in the reference laboratory of Flavivirus, which is led by the Dr. Anna Bispo, and I used to work safely with the Dr. Luis Alcantara in this research lab. So this is actually the main proposal and the main workflow that we are actually using. So we start those activities that we used to call genomic surveillance activity since 2016. So we implement for the first time the use of the nanopore sequencing in Brazil during the Zika virus outbreak, and then we extend, of course, to try to follow the track and track other emerging, circulating, co-circulating viral pathogens in Brazil. All of those activities are mainly focused on this main project and main objective, which is called now real-time genomic epidemiology, that focus in the use of genomic data generating sufficiently quickly to try to inform or trigger epidemiological intervention in investigation and outbreak control measures, of course. So actually we used to combine genomic data that are normally obtained by using next generation sequencing. So currently we are using mainly nanopore sequence, but actually we are also using Illumina, as well as, of course, ion torrent technologies which are completely available in our institute. And then we try to obtain and combine those genomic information with epidemiological and epidemiological data to try to reconstruct the history and to try to follow the spread of these new viral pathogens. Of national, of course, as well as international conference. So here I want to just show you some of our recent works that have been realized in collaboration with the Brazilian as well as Italian institutions, also including other important actors from South Africa, Oxford, as well as US, in Florida mainly. So this actually this was really our first research article that have been published by the end of January 2020. So actually we, using the phylogenetic approach, we were able to try to demonstrate that the bad SARS-like virus was the main related to the SARS, to the novel coronavirus, right? And then using homology modeling analysis, we were able to describe and identify those kind of mutations that appear to be responsible of that kind of mechanism that we used to call jump species, that gave the possibility like to jump from the bat to the final host, which is of course humans. Following this path, we actually, in collaboration with the University of Florida in Gainesville, we also try to track and to date back the circulation, the introduction of this viral pathogen in our worldwide, actually, right? So we know that the first cases start to be identified by the end of December 2019, and using those approaches, we were able to demonstrate that the virus was already circulating like in a cryptic way, in a silent cryptic way, at least 2 months before its first detection, following a path that we have already saw also, for example, in Brazil with the Zika virus, right? Well, so considering those aspects, we were also really, of course, interested to try to understand the Italian epidemic. So we provide evidence regarding the first initial sites regarding the SARS-CoV-2 transmission in our country. And then we provide, of course, by analyzing a huge amount of data, we provide insight regarding the fact that the Italian epidemic was the result following the same path that we already saw in other countries worldwide, was the results of multiple independent introduction events. So all of these considerations really and strongly highlight how the connectivity of the countries, as well as human mobility, as well as globalization, may be really responsible to mediate and modulate the introduction of new viral pathogens where they were not really circulating. Well, then we try to investigate also, to try to get more insight also regarding the pathogenesis of the virus. We try to investigate the role of some non-structural and structural protein, in this sense, the non-structural one, the SP2 and 3, and their role in the pathogenesis of the virus. So we found evidence that some mutation may be associated with, or may be related to provide like an enhanced affinity to the human host. And then in collaboration with a group of the Professor Robert Gallo in Maryland, we found for the first time evidence of mutations that apparently were associated by country and or continents, right? So all of these really allowed the possibility to identify a global and international classification system, which is actually able to identify all the possible lineages and sublineages that are circulating and co-circulating worldwide, which is called pangolin, right? Well, actually, each lineage and sublineage are defined by a specific set of mutations that can be identified in each of them. And in this sense also, we also try to find some evidence regarding the role of the NSP6, non-structural protein 6, and how some mutation apparently present in these protein could really affect autophagy. So we found evidence that some of those mutations apparently were associated to try to create like an instability of the virus into the human host. I'm really happy to share with you. So this is actually our last paper that was recently accepted. It was really such a huge effort and really such a huge work. So we try to describe the Italian epidemic, actually the 11 months of the Italian epidemic, try to characterize like, we combine actually epidemiological, of course, and genomic information to try to describe what really happened in Italy and how, for example, intervention measure really has mediated like a huge effort in trying to decrease the number of cases, right? So we saw that actually during the pre-lockdown, during the phase 1, we had like the peak of the first infection, and then we provide evidence that really phase 1, phase 2, and the beginning of phase 3 at least, really were important to evaluate the decreasing in the number of new cases, right? So actually we provide also evidence that the reduction into the non-pharmacological actually components like the reduction into the lockdown, for example, really mediate like our second epidemic wave. So highlighting again how the use of non-pharmacological components really can help to fight the our war against the SARS-CoV-2. As I said before, actually by using those genomic data, we were able also to describe the co-circulation of multiple lineages in Italy, which of them apparently appear to be really related. Of course, this is already obsolete because we know already we have co-circulation in Italy also of the variants of concern. So it is important to know that everything that we are seeing with SARS-CoV-2 is really dynamic. I want to say that everything is going to change like day by day. But the beginning, it was November 2020, December 2020, actually we're able to describe the co-circulation multiple lineages, which some of them were related of multiple introduction events that have occurred over time. So we also try to investigate the role of Italy as importer and as well as exporter. So we know that, of course, without any doubt, the Italian epidemic was the result, at least at the beginning, of some importation events, but Italy also was able and can be considered like a huge exporter for the SARS-CoV-2 also for other countries, including South America, including Brazil, considering that also the first complete genome sequence from Brazil was a returning traveler from Italy, right? Well, as I said, we were also involved to try to understand the Brazilian epidemic with our group in the Fiocruz. So we used to work also with the SEGE Lab, which is like the coordination of the reference as well as public health laboratories which are located in each Brazilian state. So we used to work with some of those LACENS, which are located in Bahia, mainly Pernambuco, which are located in northeast Brazil, as well as other ones which are located in southeast Minas Gerais, as well as in Midwest Brazil, including Mato Grosso do Sul state. During that time, it was February 2020, we received the first likely imported cases from Minas Gerais. So by using a metagenomic approach developed by the CADI project, which is an international Brazilian UK cooperation, we were able to detect the presence of, in those SARS-CoV-2 suspected sample, the presence of a rhinovirus C as well as a picobirnavirus, and we know that, of course, the first confirmed case, it was confirmed 26 February in São Paulo state, which is located in Southeast. So after the detection of the first Brazilian imported cases, we actually perform a huge work in collaboration with the Ezequiel Dias Foundation, which is located in the states of Minas Gerais, and we describe the initial insight of the SARS-CoV-2 epidemic in the states, also combining epidemiological information from the states of São Paulo and Rio de Janeiro, which with Minas Gerais are located in Southeast. and we obtain the first 40 complete genome sequence from this region. Well, actually now we are also extending those activities, as I said, working in collaboration with other labs. So we describe the early genomic detection of autochthonous SARS-CoV-2 P1 variants in Northeast Brazil by analyzing and performing an active monitoring into the states. And of course, we characterize like the genomic mutation that were, of course, lineage specific. Among them, of course, the ones that appear to be like the mutation of consent, of international consent. Despite those activities, we actually used to combine wet and dry lab, so we were also super involved also in the development of bioinformatics tool, just because we know that NGS strategies cannot be considered like a panacea, right? So the most challenging part is the bioinformatic data and analysis of generated data, which are like a huge amount. So we developed the Genome Detective in collaboration with the researcher from Belgium as well as from South Africa, which allow to realize like in nearly real time the assembling of genomic data obtained using several NGS platforms, including Nanopore, Illumina, Ion Torrent, as well as also for Sanger sequencing. Despite these, actually, the Genome Detective also include many tools which are, I can say like a typing tool, right? Including also the coronavirus typing tool, which allow in just few seconds the possibility to analyze FASTA file, wanna say consensus strain obtained from our viral pathogen, and is able like to perform for us like a preliminary phylogenetic analysis, and also describe like the kind of mutation that can be detected in our new strain. I'm really happy also to share with you our last and really recently published data from the South African also epidemic. So I'm really proud to say that I was working with Tullio in this, and also with some other amazing scientists from South Africa mainly. So we were actually I was in South Africa in Durban in March 2020. I participated like in the setup of the laboratory to try to make everything ready to receive the first SARS-CoV-2 strains. Well, actually, those are the results from our first paper, which described like the co-circulation of 16 novel variants in South Africa. And then most recently, and this work was really done during our holidays, Christmas holidays, actually we also describe the ones that actually today is considered to be one of the main variants of concern, right? Which is the 351 or 501 V2, that is the South African variants. So we describe these variants that actually is characterized by the presence of 8 specific mutations, mainly the majority of them which are located like in the RDB domain, which is mainly important to provide the affinity with the ACE2 receptor into the human host. So despite those activities, I think that we really, as I said at the beginning, during the SARS-CoV-2 epidemic, we have the possibility to realize this huge effort, and we really show with this huge cooperation effort like worldwide. So we already were able, like all together, to try to provide some evidence and some information regarding this novel pathogen. But we need to say that there are still too many facts that need really to be studied more in depth, right? So, for example, what are the pattern and driver of local, regional, international spread, which will be in turn really important for the spatio-temporal prediction, and also, for example, which is the extent of virus genetic diversity, for example, in the case of the variants, which appear to be really challenging for vaccine and diagnostic design, as well as, of course, if there are or not association between, for example, changes into the viral genome and the likelihood of COVID-19 complications. So in a sense, I'm just in the final part of my presentation, but I want to just raise 3 topics that are of particular concern in my point of view, and that are actually my main, the objective of my research in this moment, which are, first of all, what about asymptomatic cases? So we know, for example, if we can consider like the Italian epidemic, we know that the effect of our unlucky summertime was mediated also by the presence of some asymptomatic cluster that really mediated a huge amount of familiar cases, right, and community ones. So I think that the active monitoring in asymptomatic SARS-CoV-2 asymptomatic cases will be in this sense really challenging. So in this sense, in collaboration with the Professor Ambergallo, we mediated one study by analyzing asymptomatic cases, and we were able to detect the presence like of a deletion that can suggest the possibility to generate, I can say, like these differential clinical outcomes into the asymptomatic individuals. But we need to understand that actually asymptomatic people can, of course, transmit the disease. So these appear to be completely challenging, I think. Another important point, I think, that it is the SARS-CoV-2 persistence, right? So I think that actually this topic really need to be described more. So we need to understand if the virus is able to persist in different times, in different, of course, body fluid, for example. And here I just show you our really recent, also during our Christmas holidays, recently data that we have obtained in collaboration with the Professor Arnaldo Caruso and the Professor Simona Fiorentini from the University of Brescia. So we characterize and we detect, like we provide evidence of SARS-CoV-2 persistence in an immunocompromised patient, and we saw like the intra-host, we characterize the intra-host evolution. Right, so in the second episode, we saw also the presence of the N501 mutation, which is also shared in other important SARS-CoV-2 variants of concern. The last point is the problem of the SARS-CoV-2 infection, right? So we know that actually during the end of the first phase, I want to say that we were a bit happy regarding some evidence of the one that we used to know, like the achievement of the herd immunity, right? So we try to understand, we try to find some evidence regarding what really reinfection could mean for COVID-19. But actually during the last time also, because of the circulation of some variants of concern, we start to think about the false promise of herd immunity for COVID-19. So in a sense, we know that actually we need to be really careful to try to describe and identify some possible SARS-CoV-2 infection. So we need to really meet some criteria like strictly. Some of them, of course, need to be like epidemiological criteria, for example, including the period of time during which the case did not present symptoms of primary infection by SARS-CoV-2. They need to be higher than 90 days. And then, of course, a negative laboratory test from the first and the second episode. And some of them are of course, like laboratory criteria, including, for example, complete genome sequence of SARS-CoV-2 for primary and second infection, indicated that they belong to different genetic clusters and or clades or lineages, of course, regardless of the number of single nucleotide variations. So we expect the virus to mutate approximately 2 SNPs per month. In a sense, I want to share with you our last and recently data regarding the detection of true infection cases. So one of them have been described in a 35 years old female. She is a health worker, so she used to work in the San Rafael Hospital, which is located in Bahia State in Salvador. And so she reported two cases of SARS-CoV-2 infection. The first one was detected in May 26, and the second one was separated by an interval of 147 days, was confirmed on October 2016. So, of course, we use a combination of molecular screening, serological consideration, as well as next-generation sequencing to try to identify and describe the different viral origin of these two infections. So, for the diagnostic assay, we use the ALPLEX multiplex assay targeting 3 genes, the IN, E, and the RD/RDP genes. So we found evidence of the SARS-CoV. We detect the presence of SARS-CoV-2, and the CT values from these 3 genes were 25, 26, and 27. And in the second episode, again, we detect the presence of the SARS-CoV-2, and the CT values were 21, 12, and 17 for all the 3 genes that have been targeted. 4 weeks after the second episode, also the patient was EGG positive, and this was tested by chemiluminescence with the index value of 2.15. Of course, as I said, we then proceed with the next generation sequencing. So we obtained complete genome sequence with a coverage higher than 99% from the sample from the first and the second episode. And actually, we use phylogenetic analysis to try to describe the different viral origin. So in the first episode, we detect the presence of the B.1.1.33 lineage, which is one of the main lineages circulated currently in Brazil. And then in the second episode, we detect the presence of the P2 lineage, which was discovered for the first time in Southeast Brazil, in the Rio de Janeiro state, and which is considered one of the lineage of interest that is actually and currently circulating in Brazil. Well, then we try to understand what was like the spectrum of the infection in this patient in both the first and the second episode. So we found evidence that the second episode was much more severe than the first one, and this is really not so common in SARS-CoV-2. So normally the second episode, considering the literature, of course, that is currently available, is a bit mild than the first one. But actually, we were a bit worried about the detection in the second episode of the P2 variants, considering that she has also one of the main mutations that is considered of an international concern, which is the E484 key mutation. So this opens, of course, to many other questions regarding, for example, the presence of those variants, like to be able also to determine differential clinical outcomes. And then this is our really last data from another infection case that have been detected in Minas Gerais states. So the first and the second episode were separated by a 225 days, and actually we provide, of course, again, we use next generation sequencing to obtain a complete genome sequencing with a coverage of 99, actually 100% of the of those strains, and also we use phylogenetic analysis to try to describe the different viral origins. So in this case, actually both episodes were symptomatic, but the symptoms were mild. And an important thing was the detection actually of the B.1.2 lineage, which is emergent in Brazil, which is currently circulating in North America. And so actually during the last months, there was like a huge concern also regarding this variant, because there was like a huge amount of genomic data that presented like some mutation into the position 677 of the spike protein, which maybe suggests like the ability of these variants to be more transmissible, right? So the detection of this variant for the first time in Brazil in our infection case, which also carry this kind of mutation into the spike protein, really enhance the importance to try to perform and to reinforce the real-time genomic activities in Brazil, to try to describe and to try to, of course, prevent also and mitigate the dispersion of these variants that can really mediate a huge impact also in vaccination strategies programs. And also I think that actually the detection and the circulation of these novel variants in Brazil again highlight how the connectivity of the countries as well as the continent, again, human mobility as well as globalization can really mediate the dispersion of these new viral variants worldwide, right? Again, reinforcing the non-pharmaceutical component need to be really reinforced. So what we can say is what we have.
You have only 1 minute.
Okay.
30 seconds, 5 seconds, 3 seconds.
In 13 months, so I think that really, as I said, considering also the SARS-CoV-2 variants, we really need to strengthen the genomic surveillance activity. And just as a final remark, I really want to say that a global pandemic requires our effort to end it. So we really need to agree that none of us will be safe until everyone is safe. So that's all. Thank you.
Thank you, Marta, for this overview from South Africa, Italy, Brazil, comparing different situations, and I'm sure that it's going to give us a lot of inputs in order to better understand what's going on in these months. And then now I give the floor to Professor Giuseppe Lippi, full professor of clinical biochemistry at the Department of Neuroscience, Biomedicina and Movimento at the University of Verona. He's also director of the clinical chemistry and hematological laboratory at the University Hospital of Verona. The title of his talk is Epidemiology and Laboratory Diagnosis of COVID-19, the Italian Perspective. Thank you very much. And
thank you, Fabio. Dear friends and colleagues, first of all, let me warmly thank Professor Fabio Nero and the organizing committee of the Italian Embassy in Brazil for inviting me to deliver this seminar on the Italian perspective on epidemiology and laboratory diagnosis of COVID-19. This brief seminar will be divided in two parts. The format showing brief epidemiologic information on the ongoing pandemic outbreak in Italy, as some data have already been given by the former speaker, and the second part will be instead focused on the laboratory diagnostics of COVID-19 in our Italian country. The situation in Italy at the beginning of this week is summarized in this slide. An overall number of 3.5 million cases have been diagnosed since the beginning of the pandemic outbreak. The number of healed people is around 2.8 million. Those currently positive are nearly 600,000. The cumulative number of COVID-19 related deaths approximate 108,000, with a trend that does not seem to have clearly reversed so far, as you can see in the figure on the bottom left of this slide, defined by the gray line. The current evolution of the outbreak in Italy is also synthesized in this slide. As it can be seen, at least three major peaks of all healthcare indicators could be seen. The first one in spring 2020, the second between November and December 2020, and the last, which is still ongoing, in spring 2021. Importantly, we have recently demonstrated that the number and percentage of SARS-CoV-2 new daily positive tests in Italy are very good predictors of future healthcare pressure and total number of subjects quarantined, which makes laboratory testing an essential tool not only for diagnosing SARS-CoV-2 infections, but also for forecasting the societal and economical impact of COVID-19. Age and sex are the demographic characteristics that have influenced most disease progression and mortality in our country, as in many others likely. As shown in this updated analysis of Italian COVID-19 deaths published by the Istituto Superiore di Sanità, men accounted for nearly 60% of all deaths and up to 95%. Whether the higher mortality in men compared to women depends on higher prevalence of comorbidity or other biological differences is an aspect that needs to be still defined. One aspect in the pathogenicity of SARS-CoV-2 is that this virus finds fertile ground in people with one or even more comorbidities. Cumulative Italian data suggests that the death rate in people without a single comorbidity is around 3%, but then increases exponentially in those with 1, 2, up to 3 comorbidities. There seems to exist a very strict relationship between develop severe or critical forms of disease. Notably, the most frequent comorbidities in patients dying with COVID-19 in Italy are hypertension, obesity, metabolic disturbances, especially diabetes, chronic lung diseases, cardiovascular diseases, renal and gastrointestinal disorders, among others. Another important aspect that we have recently highlighted in this work is that there are some notable differences between the deaths in Italy and Brazil. Compared to Italy, we found a higher prevalence of female deaths in Brazil. Moreover, those who died in Brazil were consistently younger and had less comorbidities. The prevalence of diabetes was similar between the two countries, while that of cardiovascular, respiratory, neurologic, and renal diseases was higher in Italy. Obesity was also 3 times more frequent in patient death in Italy than in Brazil. Another study that they have recently published in collaboration with the Federal University of Paraná in Brazil found that significant increases in case rate fatality in young and middle-aged adults after identification of the novel SARS-CoV-2 P. Even if a general increase of mortality for all ages has been observed after endemic diffusion of the P.1 variant, a 3-fold higher mortality has been especially observed in patients aged between 20 and 29 years old. What has become rather clear now is that a global answer to this new pandemic disease strongly relies on laboratory medicine, whose contribution can embrace initial diagnosis, disease monitoring, and epidemiological surveillance. All these steps would be virtually unfeasible without the help of in vitro diagnostic testing. The national guidelines that we have developed for diagnosing SARS-CoV-2 infection are actually a convergence between those of the World Health Organization and the Task Force on COVID-19 of the International Federation of Clinical Chemistry and Laboratory Medicine that I have the pleasure and privilege to chair. Briefly, according to our current recommendation, routine diagnosis of SARS-CoV-2 infection shall be based on detection of unique sequences of viral RNA by nucleic acid amplification tests, especially reverse transcription polymerase chain reaction, while sequencing can be used for confirmation when this may be necessary, for example, for identifying or monitoring new or emerging SARS-CoV-2 variants. Optimal diagnostics shall then consist of an amplification test based on at least 2 independent SARS-CoV-2 gene targets. These are mostly respiratory materials and include either upper respiratory tract specimens represented by nasopharyngeal and oropharyngeal swabs, or lower respiratory tract specimens such as sputum and endotracheal aspirate or bronchoalveolar lavage, especially in patients with severe respiratory disease. We are also now considering the possible use of saliva, though we have not as yet indicated this biological material within our interim recommendations. As concerns sample pooling for increasing testing efficiency, a technical proposal has been developed based on the recommendation of the European Centre for Disease Prevention and Control. Therefore, we would here recommend that this strategy shall only be based in areas with low disease prevalence, for example, lower than 5%, shall be used for population screening, but not for diagnosing SARS-CoV-2 infection in highly suspected or symptomatic cases. The pools shall be prepared by mixing upper respiratory specimens or saliva, but not extracted RNA, validated for being used for this purpose. The methods based on the RNA extraction are globally preferred. 5 seems to be the optimal individual sample size for each pool. The presence of interfering substances, such as hemolysis or antiviral therapy, must be absolutely ruled out. An accurate, preferably automatic traceability to the individual sample that will be mixed in the pool must be assured. And finally, a second aliquot of the individual clinical specimen shall be available for individual testing when the pool screening turns out to be positive. Based on available evidence, we have also constructed a tentative algorithm for usage of rapid antigen testing in COVID-19 diagnostics. Briefly, rapid antigen-based tests may be useful for targeted population screening, as currently indicated by the WHO, or for emergency testing of subjects with COVID-19 symptoms, for rapidly detecting patients with SARS-CoV-2 infection displaying high viral loads, who incidentally are the most infected and who may need early treatment when symptomatic, or immediately timely isolation for the community. For those testing negative, the likelihood of an ongoing SARS-CoV-2 infection with high viral load is expectedly low, and thereby clinical surveillance, repeated testing, and reinforced use of preventive measures appears an overall feasible strategy. As then concerns serological testing, this has been conventionally defined as a diagnostic procedure used for detecting an immune response against an infectious agent. This definition shall then dissipate any residual doubt about the fact that this testing is not meant to replace the identification of viral RNA for diagnosing SARS-CoV-2 infection, but must rather be used for establishing whether individuals have been infected by the virus or have developed an immune response. In keeping with this, at the end of the last month, we have also published some operative recommendations for using serological testing in our clinical laboratories, as summarized in this official document. Due to time constraints, I will not be able to go through all our recommendations. However, if you are interested, the document could be downloaded for free from the website of the Italian Society of Clinical Biochemistry and Clinical Molecular Biology. A practical example on the usefulness of serological testing is in the study promoted by the Veneto region and carried out in the university hospitals of Verona and Padua, 2 areas strongly hit by the virus. According to our results, we could calculate that the SARS-CoV-2 seroconversion had occurred in 4.6% of all our healthcare workers, with higher prevalence among assistants and nurses than in physicians. Interestingly, as already confirmed in many other separate surveys, the seroprevalence rate seems lower in asymptomatic subjects, intermediate in those with mild symptoms, and higher in patients with severe illnesses. The second part of this project will now be focused on monitoring the extent and duration of the immune response, developing after widespread vaccination of the nearly 6,000 healthcare operators of our 2 hospitals in Verona. We have already obtained important findings with this follow-up study that I can only briefly anticipate, since the full article, well, is no longer available in form of submitted article because it has been accepted by just 1 hour ago. And shown in this slide, we have found the clear kinetics of all anti-SARS-CoV-2 antibodies, development and positivization, involving all immunoglobulin classes, which is magnified after administration of a second dose of the mRNA vaccine. Since monitoring post-vaccine humoral response seems unavoidable for defining the possible extent and duration of anti-SARS-CoV-2 neutralizing antibodies, on behalf of the Italian Society of Clinical Biochemistry and Clinical Molecular Biology, we have also developed an interim recommendation for this purpose. Specifically, we now advise that anti-SARS-CoV-2 antibodies, preferably anti-spike and or anti-receptor binding domain of IgG and eventually IgA classes, shall be quantified before vaccination, thus allowing to prioritize vaccination to SARS-CoV-2 seronegative subjects. We also prefer that only those immunoassays which exhibit good correlation with neutralization activity shall be adopted for this purpose. Quantitative method shall be preferred over those producing only semi-quantitative or qualitative results. Quite understandably, fully automatic techniques may be more suited when there is a need of performing large volumes of anti-SARS-CoV-2 antibody testing. Monitoring of anti-SARS-CoV-2 neutralizing antibody shall be extended for at least 6 to 8 months after vaccination, beginning 1 to 2 weeks after administration of the last effective vaccine dose, and this monitoring shall be done always using the same assay, since there is still uncertainty about assay harmonization despite availability of a new WHO international standard. Finally, anti-SARS-CoV-2 neutralizing antibodies shall be measured and monitored in patients with immunodeficiencies and cancer, in whom the immune response may differ from that of healthy people. Thank you for your attention.
Thank you very much, Professor Lippi, for this interesting talk. And I'm still thinking about this difference pattern of the mortality between Italy and Brazil, and it's important to underline the role of antibodies in the fighting against the disease. So now I invite Dr. Flavia Riccardo to close this session between molecular biology and epidemiology. The title of her talk is Data for Action from an Italian Perspective, How Epidemiology Can Support Coordinated COVID-19 Response. Please, Flavia. Flavia, it's your floor.
So, good morning, good afternoon, and thank you very much for this opportunity to be with you. My name is Flavia Riccardo. I work at the ISS, the Istituto Superiore di Sanità, and I will try to bridge from previous speakers, trying to bring the perspective of public health and applied epidemiology and how we can actually use the data and how it has been used in Italy to try to support decision makers in deciding how to respond to the outbreak. So I will see if this moves, yes. So just to open, so clearly we've discussed this before, Italy has had the unfortunate primate of being the first country detecting local transmission in Europe. And after a transition period, we're now, as Professor Lippi said, again in an acute epidemic phase, which started in October last year, well, this year, 2020. Since February 2021, we've had evidence of circulation of SARS-CoV-2 variants, as was beautifully described this in earlier talk, and currently we still have a widespread increase in transmission, and I will show also increase in prevalence of some of them, in particular the VAR 2020-12-01 variant of concern. So I will try to be fast in my talk, but I will try to bring you an overview of the different epidemic phases. So starting from what we called our first epidemic phase, more or less from February to May 2020, the transition phase from June to September 2020, where we had a very low circulation, and then a second epidemic phase, acute phase, from October 2020 and ongoing. And this is the beginning of the outbreak in Italy. So we had a moment in which we didn't really have local, as far as we know, a local circulation. But if we look at the epicurve here, we see in dark gray the cases by date of diagnosis, and in lighter gray, the cases by date of onset. And if we look at the date of onset, we clearly had a moment in which we were not detecting the presence of a local transmission, but it was there. And then, of course, we detected it and we acted in trying to respond. But surveillance had already been set up, of course, before detecting local transmission. And it was mainly around a severe respiratory infections, because this was what was known at the time. If you remember the beginning of first articles coming from China, talking about severe atypical pneumonia. And then, of course, it shifted to a more lab-based surveillance, where the laboratory confirmed diagnosis led to the identification of cases, regardless of clinical presentation. And of course, Italy had set up a number of activities to try to at least delay the arrival of this outbreak. It had declared a national health emergency very early on in the 31st of January 2020. It had, you know, at the time, of course, that the local transmission was detected, and at the end of February, a number of very quick actions were taken locally and then progressively in a wider scope to try to limit very strictly mobility and interactions among people. And then from the 4th of March last year, the first national level measures were set up, and then became a national lockdown, which progressively then tightened over the period between the 11th of March and the beginning of May. And well, this is the situation at the beginning of the outbreak. Of course, Epidata told us quite early on that this was not an equally distributed outbreak throughout the country. There was a clear increased number of cases in especially regions in the north of the
Flavia, scusami, mi stanno dicendo se puoi parlare un po' più vicino al microfono, perché c'è il suono che viene e va. Sorry.
Okay, is it better like this? Is this any better?
Prova.
Yeah, I'm getting closer. Is this better?
Okay.
Yeah. So the outbreak was not equally distributed across the country. Some parts of the countries were more severely hit, and particularly, as you can see, regions in the north of the country, especially if you look at age-adjusted attack rates, for example. And so while the south part of the country was very spared, very much spared in the first outbreak phase. Another thing that data helped us a lot with was to try to understand the impact of early measures. And you know, when we started having the first strict social distancing measures, that was on the 23rd of February. So it was like 3 days later, 3 days after the first confirmed local transmission case. And you can see that basically the transmission as inferred by the RT calculated on the date of onset of new cases actually dropped below the epidemic threshold on the 28th in the 11 municipalities, but that were first affected by those measures. This told us actually that this strategy worked, because before our experience, the only other country that had enacted this kind of measures had been China, and it was a very different context. So this was very important for us to know that the measures that were being set up were being effective in reducing transmission. So if we look at the take-home points from the very first to cope phase, well, data confirmed very early on, I would say the tremendous impact that this outbreak had in terms of overloading hospital services. I think this was quite challenging from the very start, but it also gave information on the early impact of social distancing measures. So at least we knew that what we were doing was affecting transmission. The central role of contact tracing was also precautiously recognized and implemented in a number of regions, and some examples of some of those regions and their actions has been described earlier today. Household transmission was identified as an amplifier, but it was also difficult to control it, also in lockdown conditions. And there were a number of priorities that were identified, you know, prioritize and protect fragile settings, optimize the use of diagnostics and personal protective equipment, protect healthcare workers and social workers, because data was telling us that they were disproportionately affected in the first part of the outbreak. And we also needed to remodel the healthcare system, identify legal mechanisms to support this unprecedented response, and also support strategies to enforce social cohesion. And I must say that this was something that actually happened. In the first response was very much of a synergistic response to the outbreak. We needed to be icebreakers in Europe in terms of implementing a number of tough decisions, but it was a quick and it was a coherent response. And the thing that was quite striking, I think, in Italy was the resilience of the population. There was a great level of acceptability, even though the measures that were imposed were extremely strict, even compared to what we're facing today. So I think this is an example of how then, this data has helped support that decision and at least give information on its effect. And if we look at, again, this RT, so our transmission rates over time, and we look at the time when the national lockdown was set up, and then moving on to the transition phases, we can see that at national level, the epidemic threshold dropped quite quickly below 1 towards the end of March. So from the 11th of March, when the first lockdown was set up, that was relatively fast. So it took a long time to get those cases down, of course, and incidents to go down, but transmission was dropped quite quickly. And Italy implemented a strategy which in media terms has been nicknamed the hammer and the dance, which I rather like, so I'm bringing here. The hammer is the lockdown, so a lot of stringent measures to try to bring down transmission, and then the dance, something like a modulation of those mitigation measures to try to keep the outbreak under control in the time it took to get people vaccinated, to get the vaccines, and then to have enough of a population coverage, at least to address the most vulnerable population groups. And this is where the transition phase monitoring, which I will mention, has been brought into place, which is an example of how you can use data that comes from different sources as a tool for action, a tool for decision making. So this is what the transition phase looks like. So this is where we are. We're in the summer. As was in some ways expected from a respiratory disease, we saw a drop of transmission. We went into a tired reopening. So we had a number of decrees that set out progressive reopening of activities and opportunities also of encounter, with a number of rules that were defined by a high-level national committee. But also this monitoring risk assessment process was set up at the end of April, so before the full reopening had been set up. So the transition phase, what did we learn from that? Well, it was a moment in which activities were starting to reopen again, but with a number of distancing, hygiene, and population awareness activities. Italy invested on building resilience, so it built up a lot on the healthcare system, both at public health and hospital assistance level. It tried to really implement the track and trace strategy, so really increasing testing capacity and really expanding its possibility of reaching out and identifying also asymptomatic cases, which was very difficult to do in the first phase of the outbreak, and again, enhancing a true understanding of what was going on. Transmission did not really increase above the epidemic threshold right after the lockdown. We did take some time before it reached 1 towards the second half of the summer, as I will show. Overall, however, the approach was extremely cautious and attentive. Italy had experienced a very traumatic first phase of the outbreak, and I think this also helped us to have that kind of caution in the reopening part. And also we had a number of documents, of course, that were published by WHO, by the European Commission, telling us how we should reopen and what are the aspects that should be in place. So in particular, having EPI criteria to help us monitor what was going on, trying to understand if our health system capacity was sufficient to deal with the impact of the outbreak. And of course, we need to be able to look at that in a consistent way. And this is how the idea of a monitoring tool that was aimed on one side at addressing the probability of an uncontrolled transmission, on the other, the impact of the disease on healthcare systems, on our healthcare systems. So I mean hospitals, and mainly hospital systems, so medical care and ICUs was coming up. And we use the approach to rapid risk assessments by the European Centre of Disease Control. So risk being a probability and impact combination, in which we also added an element of resilience. So in particular, thinking of public health system resilience. It's because we've seen with COVID-19, because of the nature of the disease, it takes time for people to actually go to hospital from the time they get infected. First signals of strain usually come from the public health side. I mean, people start having difficulty in tracing all the new cases, for example, earlier than we see the overload of occupancy rates, for example, in ICU. So it's just a question of time. So we added resilience as an element that could give us an earlier warning. And another thing that we did, we, of course, being this an emergency, and as every emergency, we know that data tends to suffer, especially recent data. If we look at current data, tends to be, suffers from the emergency phase, the overload of the healthcare system in general. We decided to use multiple sources of data. So we have in Italy 2 parallel surveillance systems looking at COVID-19 cases and SARS-CoV-2 infection. So we have the aggregated systems that were shown by Professor Lippi earlier, coordinated by the Ministry of Health, and the system, case-based system coordinated by the Institute of Public Health. And then we have data that's coming from the hospital monitoring services, from the Ministry of Health, and indicators that are provided by the 21 regions and autonomous provinces. And we developed a system by which we engage every week. We consolidate the data, we engage, and we publish data using this risk-based approach. Again, in the summer, in August, we looked at what was going to happen, what was likely going to happen in autumn. We all expected an increase in cases. So we looked at different scenarios based on our public health capacity and, how long we would be able to hold, for example, an increased transmission of different magnitudes. And this then all came together in a strategy that was published in early October, linking the level of risk and the actions described in the different possible scenarios that could take place and modulate over time. So in the transition phase, of course, we improved our data completeness. We had a lot more testing capacity. We didn't have uncontrolled transmission. So there was no overburdening of healthcare facilities. And we were able to roll out this monitoring tool in a relatively quiet moment. We also had increasing resilience, increased screenings, increased contact tracing. We had a lot more asymptomatics being detected for that reason. Almost all new cases were fully investigated and traced with contacts, and human resources were radically increased in terms of contact tracing, monitoring, testing to at least 1 per 100,000 population in each region. And it was really a time in which we invested in preparedness, which was what Professor Brusoferro mentioned at the opening of the webinar. And this is where we are now. We are now in the acute phase. So we had then an increase in transmission, actually later than other European countries. Ours occurred in October, actually. In some countries, you've seen this increase already from the end of August and September. And this is just to let you see how the risk monitoring works. So see, every line here is in this colorful chart is a region. And we have in green the weeks in which the regions are at low risk, in light orange at moderate risk, in red in high risk, and in dark red was when they had issues with the data quality, and therefore they were classified at high risk because of data quality aspects. Of course, when you have so much cases that you cannot reach a minimum data quality, that's already a symptom of strain. By the beginning of this, so you can see there's this shift progressively from green to light yellow over the summer. And we can see how then this becomes a darker yellow. That's when we started looking not only at moderate risk, but we looked at the projections of hospital occupancy rates at the observed transmission. So we had regions that were at moderate risk, but because of the projections, we knew that they had a high probability of moving to high risk in the subsequent weeks. And as you can see, then this allowed us to really track when the new acute phase started in Italy. This is the transmission rate, again, looking from June to December last year. And you can see how then progressively the transition phase led to a gradual increase of the RT that stationated around 1 until August. At that time, so the risk was starting to shift to yellow. So there were a number of activities made, especially looking outside, because we started to see a peak of imported cases during that time because people were returning from the holidays. And so we had quite a lot of imported cases. In October, this led to a number of screenings being done from people returning from high incidence countries. And then we were able, thanks to the risk monitoring, to issue an alert in September and to declare the change in the epidemic phase. The additional measures then will set up first more at regional level in the most affected areas, then at national, subnational level from early November. And this is when the RT drops, and then slowly over time, then this led to a de-escalation of measures. So we tried really to implement the dance, trying to close but allow also the reopening, gradual reopening. It was quite clear that a long-term lockdown was not sustainable on the long run. So we need to find a way to control this outbreak, limiting the damage, but both the health damage, but also the damage on society and the economy. Another thing that was advantageous of this method is it allows us to track, for example, what incidence was there at national level before we changed our phase. And so we've seen that, you know, when cases in Italy were around, rose above 50 cases per 100,000, that was when basically the whole country moved from control to mitigation. And this is a threshold that was then confirmed also through mathematical modeling, and I'm just leaving here the references to our paper on this. And another thing that we were able to do was to look at what incidents most of our regions experienced issues in terms of overload of occupancy rates that we set at 30% for ICU and 40% for medical care, non-critical medical care, and in 7 days. And we saw that that threshold was, I'll be fast. So it was 250. And interestingly, it's the same threshold that was set by the European Council in February this year for the highest level mitigation of mobility. So what the measures were that were set up in Italy were a combination of a number of elements based on RT, on the risk assessment, and since January on the 7-day incidence looking at these thresholds. And if you're curious, there's a paper there looking at the impact of those measures. We've seen, especially in November, that this was actually quite well balanced. So this yellow color applied to measures that were basically bringing the RT around 1. So they were sort of keeping the outbreak stable. And then we had the orange and the red, which were associated with tired measures that led to a decrease in transmission over time. But then things changed, of course, and since December, we've seen that the virus changed in ways that is of public health concern. So we've seen, I think, a beautiful presentation of how this virus changes all the time, but then sometimes it changes in ways we don't like. And in particular, we've done now 3 flash surveys in Italy looking at prevalence of variants. And as you can see, the so-called UK variant, the VOC 2020-1201, is become prevalent, from national prevalence around 18% in February. We're now 18th of March at 86.7%. So we can say now that's the prevalent variable. Of course, that's accompanied to increased transmission, most likely also severity. So it's of concern in terms of the management of the outbreak, but we also have the presence of variant 501 YV2 and P1, albeit at very low prevalence rates. We've had clusters and this presence in some regions with increasing and decreasing prevalence according to the period in time. So definitely these are aspects that we're looking into, and I totally agree with what was said before regarding the need to increase surveillance in terms of virus circulation. Finally, vaccination, that's another aspect that is predominant. Just to be really fast, in Italy, of course, as in other countries, we're boosting the vaccination process. Vaccines that are being distributed in Italy are Pfizer, BioNTech, Moderna, and AstraZeneca. These are the data from this morning. And you can see that main targets in Italy have been over 80s and healthcare workers, and now opening up to the other age groups and professions. And this is starting to show, if we look at the median age and first hospital admission ICU, we're starting to see a decrease in the median age, which is interesting. Also, we've seen a decrease in the proportion of healthcare workers on the left. And for the first time in Italy, we've had actually the weekly incidence of people 60 to 79 years has been higher than the over 80s, which hadn't happened before. So we're starting to see some timid specificatory effect of vaccination. And this is where we are now. Concluding, this is our final 2 slides, I promise, Fabio. So this is the outbreak where we are. So I hope I've shown you how data is actually helping us understand how the epidemic is going on from an epidemiological perspective. This is the risk monitoring system. We can see how, as Professor Lippi said, of course, there are different moments in this acute phase in which things go worse, and we've seen it in October. We've seen it in December, even though we've had quite strict measures over Christmas. I think this helped us, and now we've seen it again in the most more recent weeks. So this is data as of last Saturday. And then in conclusion, so of course we had our outbreak start quite abruptly in the context already of a widespread local circulation. The lockdown was effective in containing the first wave, and I think we showed with data that this is the case. Transition helped us to prepare better. And of course, there are a number of tools that are being developed that are helping us to keep this dance and trying to keep the balance with activities and actions of mitigation. Mass vaccination, we have initial evidence of some of the impact, but we still, of course, have to vaccinate a lot before we will have a more clear effect. And there is a lot of work being done to strengthen surveillance of viral variants. And I'm leaving also the contact of Professor Stefanelli, who's guiding this aspect for the Institute of Public Health. So there's, of course, there are challenges. The challenges related to the higher transmission of some variants, but mostly of the immune escape aspects. And I think it's very interesting what was shown about reinfection when immune escape variants are present, because we cannot expect milder disease. We expect a new infection without any maybe immunological progression, and the modulation of measures, of course, and vaccine coverage in terms of what this will mean for vaccinated people and measures there. So thank you very much for your time, and I'm leaving some context for any additional questions. Thanks.
Thank you very much, Dr. Riccardo. It's an excellent presentation. I'm sorry that sometimes time runs so fast. And I give the floor to the first speaker of a new session, Dr. Lanini, who is the ricercatore medico at the Department of Epidemiology and Preclinical Research at the National Institute for Infectious Disease, Lazzaro Spallanzani, which is the reference center for infectious disease in Italy. Please, Simone, the floor is for you.
Hello, can you hear me?
We can hear you.
Okay, so I'm going to share my screen. Okay. Okay, does it work? Okay.
It works, it works.
Okay, thank you very much. This is an honor and a pleasure for me to participate to this conference. As Professor Naro said, I'm a medical investigator at the Preclinical Research Unit at Lazzaro Spallanzani in Rome, which is an institute that is established in 1936 and since 1999, as you may note, has been recognized by the Italian Minister of Health and designed as the largest Italian hospital completely dedicated for infectious disease. We have a BLS4, a biosafety level 4 biofacility and biorepository. And as you might know, we can manage from a clinical point of view and also carry out research on patients with basically any kind of transmissible disease. So this is, for those who never come to Rome, this is my institute. Here you can see the main building, the hospital, in which we can host about little less than 300 infectious disease patients. And this is the new unit that we have opened with COVID, which now currently hosts about 60 beds for a highly infectious patient who need the critical care, and also host the new unit, the new phase 1 unit that is now completely dedicated for developing new intervention against COVID from phase 1 to phase 3. So as you may see, Spallanzani was directly involved in the Italian effort, in the national effort against COVID since the very beginning. We housed the first 2 patients. And as you may see, also our efforts in terms of care delivery and patients recovery was basically identical to that we see. Completely reflect the effort at national level. As you can see here, you can see the incident case in Italy. This is the regional case in the area around Rome that is directly served by Spallanzani, and this is our effort in terms of patients recovery. But as I told you, Spallanzani is not only an hospital, it's a clinical research center, and basically what I now try to develop, our effort is basically toward 2 fields of investigation to find out new intervention against COVID-19, vaccine and monoclonal antibody. Several of these projects I direct coordinate now, and I'm going to speak about that. So as you know, there is now hundreds of vaccinations that are being developed against COVID, and several of that are also being approved. In Italy, we actually have approved 3 different vaccinations, and we're going to, and you're waiting for the 4th one. But Spallanzani not only provide this kind of vaccination, we're also involved in 2 research for developing 2 different vaccines. Today's vaccine against COVID and also against other infectious disease in the vaccine like those that we use for Salk, anti-polio, anti-flu, or hepatitis B, or HPV. We also, vaccine has been also developed as a live attenuated vaccination, but what is important now for COVID is that the new platform, and in particular genetic platform, has been developed for new vaccination. This kind of vaccination has been firstly developed against Ebola virus, and in fact the only approved vaccine against Ebola virus is through this new kind of vaccination, genetic vaccine. But also all the new anti-COVID vaccination that has been approved in Italy are basically developed on genetic vaccine platform. But what are actually genetic vaccine? We have 2 main families of genetic vaccination. The first of them are those that we call vectorial vaccine. A vectorial vaccine is basically a viruses that we have in
Simone, I cannot hear you. I think that the band is overloaded. I suggest you to switch off the video.
Sorry?
Okay?
Okay. Fabio, va bene?
Okay, now it's okay.
Okay. Sorry, it's a helicopter. Okay, the second way for developing genetic vaccine is that is through RNA vaccination, which is something which is the way in which Pfizer and Moderna has been developed, and in this way we can introduce into the cytoplasm of the cell of the host cell the information for producing the viral proteins that eventually they will elicit immune response. And another way is through electroporation, which can, in this way, we can transfer directly into the cellular nucleus a DNA plasmid, which actually can elicit, promote the production of protein and eventually the cellular response. But basically, the main difference that we have from all kinds of vaccination such as those performed antigen and genetic vaccination, is that in genetic vaccination, the antigen is produced inside the cell, and so that the immune response follow the natural pattern of infection, and in theory is much stronger because it is mediated both by the major compatibility complex, the type 1 and type 2. So Spallanzani is currently developing two different genetic vaccination. One is a vectoral one, which I'm going to speak about, and the second one is in the very earliest phases. I'm not going to speak about it, but it's a DNA vaccination, which we are going to develop with Tages. This is the preliminary data of our phase 1 vaccination, first one trial on a vectoral vaccine, which we developed with Reitera. This is a vectoral vaccine that has been developed through the engineerization of a gorilla adenovirus, in which we include the genetic information for the spike protein. Currently, we have already finished the phase 1, but what I want to discuss with you is that in that phase 1 study, we found an extraordinarily good profile of safety. We had no severe reaction, and we also have not serious reaction. And despite that, the antibody response that we observed was very strong at all the doses that we use, and most importantly, we have a very good production of neutralizing antibody. As we expected, also the cellular response was extraordinary, and in fact we see that was strong and completely shift towards a Th1 response, which is the exact response that we want for fighting against respiratory infections such as COVID-19. And in fact, thanks to this good result, since March 10th, we have already started the phase 2/3 trial, which is currently ongoing. The second challenge that we take up was that to develop new monoclonal antibody which are directly against the SARS-CoV-2. And basically, monoclonal antibody are the copy of those neutralizing antibody that the human body produce to fight the infection. But thanks to molecular engineering, we can purify them, have a pure formulation. We can engineerize the molecular structure in order to make them safer than other antibody, and also to optimize the pharmacokinetic, and also for better production, and monoclonal antibody production can be scaled up into an industrial process. Today, we have in Italy approved 2 different kind of monoclonal antibody, which we already are providing to our patients as Spallanzani. But in addition to providing antibody, a therapy that are already approved, we have also started the development with Toscana Life Science, which is an Italian foundation dedicated to science, for a new monoclonal antibody, which has the characteristic to be very potent and also to have an optimal pharmacokinetic profile that allow us to administer to patient this monoclonal antibody by intramuscular injection. Basic limitation for monoclonal antibodies are quite well known right now. There is no benefit for patients with severe and with severe and critical COVID, and also the costs are quite high. But there is important perspective for the development of monoclonal antibody for early therapy of patients in case of post-exposure, and also as alternative for those patients who cannot receive a vaccination, either because they are severely immunodepressed and or because they have a severe reaction in the first vaccination. The Italian monoclonal antibody has been already passed through the preclinical phases, and data has been already published. I just want to show you what we have done right now. We basically select the antibodies from Italian patients here at Spallanzani. Then from patients, we select the cells, the B cells that are able to produce this antibody, this natural antibody, the polyclonal antibody. Then we select the strongest one, and then we engineerize their structure in order to make them longer, more persistent in plasma, and also to be less reactive. And eventually we start animal challenging. We have already completed animal challenging, and we have completed animal challenging using the golden hamster. This nice animal is in fact one of the best models for pneumonia, for COVID pneumonia. And we challenge this animal, and we use this animal to model either a therapy or a prophylaxis. And what we found is that in both circumstances, this monoclonal antibody has a clear effect in preventing infection, as you can see here. The animal do not, sorry, not infection, but the disease. As you see here, we use the body weight variation as the proxy of the COVID disease. And as you can see here, the administration of the monoclonal antibody completely prevent the infection, the disease, sorry, not the infection, the disease in our animal model, and also can improve the recovery in animal which has already infected and already have a disease. But most importantly, what we see in our experiment is that this monoclonal antibody not only can prevent the disease, but in the prophylaxis experiment can completely prevent the viral replication into the lung, and also after infection can completely remove viruses into from the hamster's lungs by the third day after the administration, suggesting that the same pattern might also be observed in human. And in fact, we are currently on phase 1, and and today I have enrolled the 20th patients in our trial. So, what can I see from our perspective, from the perspective of a clinical researcher, in terms of what we can actually do with new research for intervention against COVID? COVID will be endemized, or we will be able in the next future to completely eradicate the infection. But there is something that suggests that in fact COVID may become endemic, because the immunity is waning. We know that it's rather heterogeneous across people, across different population, and in fact we have also reported the first cases of infection, and also most importantly we have also variants. The virus can vary and escape the immune system, and even potentially even vaccination and monoclonal antibodies. But research is going on, and we will have, hopefully in the next future, new vaccine, even stronger than the vaccine that we have now. Potentially, we can also have vaccination that can be administered through nose or orally, that can be foster a local immunity such as IgA and local lymphocytes that can actually prevent infection, completely prevent infection of upper respiratory tract. And we also can have monoclonal antibodies. Monoclonal antibodies that, as I show you, can be used both for therapy or prophylaxis. So, and this make us hopefully and this make possible a complete eradication of this scourge of these viruses. So thank you very much. What I can suggest to all of you, what I think is that we must be optimistic in what we have already done, and we must be strict to evidence, to scientific evidence, and solid, and to produce solid study in order to do not betray all the trust that we have already from the scientific community and all the people around the world. Thank you very much.
Thank you very much, Simone. And I just want to, I remember the fine words that the first Chinese patients who were isolated in your hospital said when we were cured. They said that they were treated like to be in a family. So this combination of personal care and scientific attitude, it makes your institute really strong. But just one word, they faced you and free. How is it going? Well or down or bad? About the vaccine.
The Reitera one.
The Reitera one, yeah.
Yeah, it's very good.
Can you give us just in the...
Yeah, very good. We are
very
quite
good.
Close. Yeah, very good.
Perfect.
We are quite close in phase 2, and we hope to start phase 3 in perhaps 2 months.
Very good. Thank you very much, Simone. And now I give the floor to Professor Francesco Paolucci, Professor of Health and Economics and Policy at the Faculty of Business of Law at the University of Newcastle, but he also is a professor at the School of Economics and Management at the University of Bologna. He's going to share his presentation with a PhD fellow, Marcello Antonini, and they are sorry, but because of the time zone between Brazil and Australia, they cannot be with us, but they recorded their speech, so we are going to listen to what they are going to say, making an interesting comparison between Brazil and Italy. Okay, thank you very much.
Good day, everyone. My name is Francesco Paolucci. I'm a professor of health economics at the University of Bologna, Italy, and the University of Newcastle in Australia. Thanks for the invitation. Of course, we are more than delighted to present the findings of a special issue we conducted with a number of colleagues around the world, covering virtually every continent and countries within them during the pandemic, over 20 countries with the involvement of over 40 institutions and academics. This is the paper version, very old-fashioned, but you can order it in Health Policy and Technology, or find it online. We will have, of course, the links, although we really like the booklet, so we felt it was our obligation to show it to you. Okay, so the special issue that we worked on for a number of months and that we're continuing working on with a potential follow-up later this year as pretty much covered, as I said, a number of countries in almost every continent. I say every continent because we have also added papers from Africa, which was initially sort of, I would say, exposed to COVID in slightly later stages. So you can, of course, find these materials online. Towards the end of the presentation of Marcello, I will take the word again and we'll cover those practical issues. We wrote this work together with colleagues from Chile, Brazil, and Portugal, in fact. And in fact, we based this collaboration on previous ones that are long-standing, in particular with our Brazilian colleagues, Professor Menezes, and Chilean colleagues, Maria Alejandra Benitez, Velasco, and Enriquez. We wrote our paper trying to understand really how the response in 5 Latin American countries has been to try and contain the COVID pandemic. So the agenda for today is to have a short introduction about the study, clearly look at the characteristics of the countries involved, which are Brazil clearly, but also Peru, Chile, Colombia, and Ecuador. Try to make sense of the responses to the pandemic and some concluding remarks. What is it that we've done? Obviously, to understand the pandemic in Brazil and other Latin American countries, we have to have clarity about what are the pre-pandemic characteristics of these countries. And only if we do that, we can understand the mitigation and containment measures and their effectiveness. In fact, we also ran last year a number of workshops with the Australian National University with guests from Brazil and other Latin American countries to really pin down what has been the impact also of technology and intervention and also economic support strategies to respond to COVID. The 5 countries that were selected were conveniently selected so to cover the widest possible range of socioeconomic characteristics and demographic characteristics, but also to have a range wide enough of healthcare system and coverage characteristics together with development indicators in Latin America, which could then be representative also for other countries with similar futures globally. The approach that we took has been quite thorough in the sense that we conducted extensive documentary analysis. We focused on policies both at the national and the state and regional level and clearly analyzed these policies in relation to existing economic performance indicators and also epidemiological data available at the various governments levels. Now, to start understanding the picture in Latin America and Brazil and other countries in that context, we have clearly to understand the overall health and development profile. And I would say that from this slide, what we have to take home is basically a synthetic interpretation of the statistics that we have taken into account that really show a picture that across the board signals a high level of informality, both in terms of labor demand and supply. And clearly that is strongly linked to underlying socioeconomic conditions and high poverty rates. Obviously, as often this happens, the geographical result of informality and poverty typically tends to be correlated with high density, and with that also with a high demand for mobility. So this is important in the context of containment measure. Obviously, what is also striking, and perhaps Chile is the only one that is clearly trying to move away or has been moving away from this, it's the low sanitation index levels that are clearly in need of work. And I would say that the pandemic, as we will see later on, have addressed some of these concerning health and development profiles issues. Now, to complete the picture in terms of the societal view, also we need to understand the healthcare context. And here in Brazil and other Latin American countries, there is clearly some significant degree of variability in how societies have decided to organize their healthcare financing and healthcare services provision. What we can see clearly and distinctively is that the countries that have had a long-standing stability in terms of organizational future of their healthcare system with either public-private system like the Chilean system and or the competitive social insurance market as Colombia has gradually reformed in the last decades. The other countries have a default mechanism, which is typically public and public-private partnership based. However, although the percentage of coverage apart from Ecuador are pretty high, we observe that in fact the delivery compared to other countries, both in the OECD but also in this group like Colombia and Chile, show low number of physicians per population, per 100,000 people, and clearly that is reflected also in other health professionals which are below OECD average, and clearly that is a reflection also of the ability of healthcare services to have formal hospital beds and clearly also ICU beds and thereby ventilators, which are critical in the context of COVID. Now, clearly, the picture when it comes to the underlying risk factors in the population, which are important determinants to determine the severity or expected severity of COVID cases, and also in terms of understanding the presentation of these cases and the likelihood to lead to mortality or death, is fundamental. And we can see here that the picture is quite complex and different from what we have observed in Italy and other developed countries in terms not only of life expectancy at birth, which is clearly approximating what developed nations display, but clearly we have a somewhat two epidemiological trends that occur, some that are still embedded and characterized and partly captured by mortality in countries that are developing, so where infectious diseases apart from COVID still exist, but increasingly a picture that we see in other countries that certainly has created a similar situation when it comes to likelihood of mortality to occur. And clearly those are epidemiological features of developed countries, so prevalence of high risk factors that relate to lifestyles such as obesity and smoking and alcohol consumption and so on. Obviously, all this now in our study has been related or correlated to a number of points of interest to try and make sense of the responses to the pandemic, that we try to understand fundamentally how the responses really link to the picture that these countries, Brazil and other Latin American countries, had in front of them at the time of the pandemic. And this, of course, has been also evaluated over time during the pandemic. So the 3 points of interest that we looked at in trying to explain the pandemic response vis-a-vis the characteristic of each of the 3 countries are whether and to what extent the timing and stringency of the response measures were adequate, both in terms of mitigation of and containment of the pandemic itself, and also in terms of economic measures to support, in fact, the large part of the population that is indeed exposed to economic hardship. Secondly, we looked at compliance vis-a-vis the mitigation and containment measures. And what we looked at in terms of metrics have been mobility, also pandemic management program, and how it related, of course, and differentiated towards the socioeconomic aspects and context underlying the Brazilian and other Latin American population. And finally, the healthcare system response. So whether and to what extent the health system has been able to have a response, and what a response, and whether that has had any impact, and that not only in terms of beds or number of doctors and so on, or ventilators and so on, but also in terms of prevention, so testing and tracing and the like. Now, what we can see by looking at the timing and stringency of the country measures is that if we look at last year, all the current countries under observation really intervened pretty early on. You can see that between the first case and lockdown, there are a number of other dimensions that are important, that signal not only the incidence of the disease, but also the responses adopted. And we can clearly observe that most countries intervene hardly and quite soon in the manifestation of COVID in Latin America. So stringency has been quite immediate, and surprisingly so, and differently from countries that we are not talking about today, like New Zealand and Australia, where we are in the moment, at the moment, which had a similar quick and fast response to COVID, but have kept this hard, let's say, response throughout the year up to now, and at the same time have seen good results. In fact, COVID hardly exists here in Australia or in New Zealand. In Latin America, unfortunately, the picture is very different, and Brazil is clearly showing pretty staggering figures. Despite the containment stringency measures adopted, cases kept going up. There have been some variation, but apart from Chile, really, in Latin America, we've seen a pretty daunting picture throughout, signaling that something is not working in terms of containment measures. At the same time, this needs to be put in the context of the underlying pre-pandemic context. And clearly, we need to find an explanation because of the failure of this measure. And clearly, one can relate it to the timing of the measures. So perhaps too much too soon is what we can take from this, but looking at the stringency measures and the cases which kept going up, we have to try and look for more in-depth explanations of it. Clearly, a lot of it is related to what we refer to as mobility, and that is clearly highly correlated with compliance to the measures in general population. And clearly, this has a very strong socioeconomic component. In fact, most of the countries, despite the attempts to limit movement, have significantly found this issue relatively impossible to maintain or even to introduce or have over time. Apart from Chile, where you had a staggering reduction, but that is not equally distributed in different socioeconomic groups. Clearly, poverty and the need to generate income on a daily basis without stable measures that hit formal economies or formal markets has hindered the ability of these countries to contain mobility. And clearly, mobility in a high-density context has made almost ineffective and certainly not cost-effective both containment measures and economic measures to try and respond to the crisis. Some of the issues with compliance, certainly in our work, has been or can be attributed to the conflictual nature of the messaging from public governments. And by public governments, I really mean the different levels of government, with in Brazil in particular quite a bit of variation on the messaging that led really to a clear mismanagement that has reflected in many areas and also in critical changes at the government level, with the information really differing from the local health authorities level to the central government levels. Now, it is important also to recognize something that is of fundamental importance. Clearly, more for some countries like Chile and Peru and Brazil and others, that really has transformed the healthcare system. Like discussions around universality of healthcare, apart from the right to access the limited amount of care, have changed completely nature in some countries. For example, Chile, where we can say that the levels of ICU beds and hospital beds and also workforce have really been boosted up. And this has been a trend also in the rest of the Latin American countries and also in Brazil, although the scale has been much smaller than other countries due to constrained resources and political decisions. Now, I would say that that has not been true for all the aspects that you would want health systems to be responsive to. Clearly, as much as we've seen in terms of healthcare provision relative to COVID, we have seen a response, positive response in investment that has not been really detected in other very important elements, and this is not dissimilar to other countries, including Italy, where de facto tracing in particular hasn't really worked as well, at least as it has worked in Australia. And clearly the strength of policy responses and measures needs to be accompanied with a strong prevention, tracing, organizational feature in the response to COVID to have and optimize the results as New Zealand and Australia have been able to achieve compared to Brazil, clearly, and other countries. Now, to conclude, all 5 countries have been intervening harshly and soon. They have maintained a dynamic response with the dynamic lockdowns and curfews throughout the period. Obviously, that as the effectiveness has been of this measure has been affected strongly by the underlying fragility of the healthcare system and by the clear lack of resources. But not only, there are underlying disparities that have little to do with healthcare, but more with health, income, education, and other important underlying inequalities in Brazilian and other Latin American societies, which have made any containment measure and economic support less effective than they could have been in presence of a larger, more formal underlying economy and a stronger and well-served preventative and healthcare response and healthcare system response. I would say that the response, however, has been encouraging and certainly positive in terms of investments throughout the pandemic. But clearly, the key in this pandemic compared to other countries that are still struggling, including Italy itself, as Marcello will now discuss and present, depends very much on our ability to respond very fast, very effectively and even in a hard way to be able to contain the spread of the disease soon, coadjuvated by a strong tracing and surveillance support system, and with adequate income support that were very timid and very delayed, which affected compliance. And we've seen the same in many European countries when it comes to low socioeconomic groups. Thank you very much for your attention.
Thank you, Professor Paolucci, and good day, everyone. I'm delighted to be part of this very interesting webinar. Unfortunately, I cannot participate live, but it's a pleasure for me to present this research that we did during our special issue. And first of all, I would like to acknowledge my co-authors that work with me for this research, particularly Chiara Berardi and Mesfin Geni, and also, of course, Giovanni Cotugno, Alessandro Lanteri, Adrian Melia, and Professor Francesco Paolucci. So I'm going to give a brief overview of the COVID-19 pandemic in Italy and the major intervention implemented by the government to face and handle the pandemic during the first and the second waves. Let's start to give some key points of the Italian experience. Let's remember that Italy was the first Western country to be severely affected by the pandemic, and also the first one to overtake China in the said ranking of number of cases and number of deaths. The magnitude of the COVID-19 pandemic required a huge injection of resources, financial resources, in the system, not only the hospital but also the economic one. In particular, for the healthcare system, we have noticed an increase in the public health expenditure compared to the previous year, and these investments have been directed toward the reorganization of the hospital systems and the recruiting of health personnel, but also in the purchase of equipment. You can think about the hospital beds, masks, ventilators, etc. Another important element of the Italian experience is the spread of the virus. During the first wave and the beginning of the second wave, we have seen that the virus was more spread toward the northern regions compared to the central and the southern one. Currently, this heterogeneity is much more reduced, and we can say that the virus is spread more homogeneously throughout the country. A key element to understand the COVID-19 in Italy is the response strategy is the design of our healthcare system. The Italian healthcare system is a decentralized national healthcare system where each region is responsible for the provision and the financing of the healthcare services, but also for the governance of the system. So you can imagine how this applied, like this system applied to all the 20 regions, and you can easily imagine that this model led to the coexistence of many different models in our country. So we range from the quasi-integrated model in Emilia-Romagna, for example, to the quasi-market model in Lombardy. And of course, this great variety of models and design has played a central role in the ability of the regions to cope with and adapt to the COVID-19 emergency. So here I reported the epidemiological trend that we have assisted, that we have observed during the special issue and during our analysis. I reported here 4 main panels. You can see that I divided them by geographical areas. And I reported the number of new cases, the number of new recovery cases per day, and the number of deaths per day. You can easily see that the heterogeneity in the spread of the virus is quite clear during the first wave, particularly for northern regions. You see that the numbers are quite higher than the central and the southern regions. Let's focus on the first wave. You can see that the peak of the contagion, so the daily contagion, has occurred the 21st March with approximately 11 cases per 100,000 population, and the peak of death was recorded exactly 1 week later. And you can see, maybe here it's easier to detect this in the northern regions panel. You can see that there is an increase for the gray line. So this was the peak of the deaths. After the decrease of the number of cases and hospitalization in the first wave, you can see that the government started the exit phase strategy, and then we have assisted to a blank time in which no interventions were implemented to face the pandemics, until, of course, the new cases were being increasing, and so new restrictions were applied accordingly. So to have a more, let's say, detailed description of the timing and the stringency of the interventions, I reported here a timeline, including also the seasons, because the seasonality factor seems to cover an important role in the ability of the policies to tackle and stop the spread of the virus. So what we observed is that the Italian government has for sure had a quite responsive strategy as soon as the first cases were identified in Lombardy and Veneto. You can see that there are a lot of intervention in a short period of time here, but these interventions were not enough to stop the spread of the virus. We know now, of course, that the virus was already circulating around the country, but at the time we were not sure about it. So these interventions were clearly not enough. And this inability led the government to impose the national lockdown on the 9th March. You can see here the Io Resto a Casa decree was the intervention. After the national lockdown, harsher measures were then implemented the 22nd March. And if you remember the slides that I presented you before, where there was the peak of contagion for the first wave, the peak of new cases was exactly the day before the Chiudi Italia decree. And the Chiudi Italia decree basically implemented the shutdown of all non-essential economic activities on top of the national lockdown for the population. This lockdown lasts for 2 months, so one of the strictest and strictest lockdown that we recorded in our special issue. Only after the reduction of the number of cases and hospitalizations, the government started the lifting of the restrictions in the end of April, beginning of May, and with the aim to lift all the restrictions the 3rd of June. You can see that after the lifting of the restrictions, there is a gap during the summer that I showed you also before in terms of interventions. The first one was recorded the 16th of August, in which the government implemented the closures of discos and nightclubs, and also implemented the requirement of wearing masks in public spaces. That's, of course, one of the main mistakes that has been done in the response strategy, and probably we are still paying the price for this lack of intervention. This was the time in which the coronavirus seemed to be disappeared. It was not, clearly it was not disappeared. Anyway, after the closure of discos, we have still a moment in which, like a period of time, a quite long period of time in which the government was not sure whether to implement stricter and harsher measures. Indeed, we had to wait until the 24th October to a new restriction, and was like the implementation of the curfew for the population. So after 10 p.m. no one could go out from their house. And then with the increase of the number of cases and number of hospitalization, etc., there was a new decree implemented that was about the introduction of the so-called differentiated lockdown, the 4th November. We have still this kind of response strategy. And here I report briefly what it's about. So all the regions are assessed in terms of their risk. The higher risk is the red color, while the lowest is the white. And then here we have yellow, orange, and red, of course. So the differentiated lockdown shifted the responsibilities from the central government to the regional governors. This was already happened for the lifting and for the exit phase, but now the responsibility for the regions is much higher. The Ministry of Health, however, is still responsible for the assessment of the risk of each regions, and particularly is responsible to assess the 21 parameters that are used to assess the risk. These 21 parameters are interesting to notice, because it was the first time that we have a clear information about what were the metrics used by the government and the advisory board to assess the risks. Of course, the usual suspects are included, such as the number of symptomatic cases, the number of hospitalizations, the transmission rate, etc. But what we found in our analysis is that the number of hospitalization has really been the driver for the response strategy. So in this table, I reported the number of hospitalization over time and also the response strategy. So you can see that as soon as the number of hospitalization increased, also harsher measures were implemented, and the opposite, of course, for a reduction in the number of cases. But the number of hospitalization was also the main weakness of the Italian response strategy, and particularly the pre-existing condition that we faced in some, and mainly in most of the hospital systems, in most of the regional hospital systems, because as you can see here in this left-hand panel, without the introduction of additional beds for the ICU, many regions, particularly the northern regions, would have seen a collapse of their system. Even Lombardy, with the addition of the new ICU beds, was really close to reach the 100%, so the saturation, the complete saturation of the system. To overcome this really worrying situation, in May, the government introduced a decree in which imposed to all the regions to achieve the target of 14 beds per 100,000 inhabitants. And these are the data for March this year. So we can see that now most of the regions hit the target, except the 4 regions, Molise, Sardinia, Campania, and Calabria. But what is interesting to notice is that when we did our analysis, until the end of November, only a few regions were able to hit this target. So fortunately, now most of the regions were able to solve this issue and to have like an adequate number of beds and capacity. To conclude the brief description of the analysis we did so far, I want just to mention the economic measures implemented by the government. We have assisted to really unprecedented economic stimulus to face the pandemic and to try to avoid bad and severe economic downturn. We can mention, for example, the Cura Italia decree and the Decreto Liquidità, that were the two main measures implemented at the beginning of the pandemic. And also it's worth to mention the European Union contribution that for the first time suspended the fiscal compact. And this is very important because even for the Great Recession, this was not suspended. So this is a signal of the new directions that the European Union wants to follow for the next crisis. So to conclude, we have assisted to a really severe impact of the COVID-19 in Italy, not only on health outcomes, but also on social and economic outcomes. The timing and the strictness of the interventions has been quite responsive at the beginning, not enough to stop the spread of the virus and to limit the negative consequences and events that we have seen so far. Main mistakes, of course, concern, in our opinion, the gap that we have assisted in the summer months. And another important point to mention is the not willingness of the government to invest in a good tracing technology, high quality tracing technology. The contact tracing app, for example, was developed and investment were made by a private company in agreement with the government, but again, by a private company, and the success was not the one we were expecting. So for further research, I want just to mention that it is important to carefully look at the effect of isolation, social distancing, and financial loss on mental health and stress levels. Particularly, this will be the main concern for the population in the long run. We have already assisted to an increase in the suicide compared to 2019, so it will be really important for policymakers and the government to look at these metrics and try to support the Italian population. And also important aspect to consider is the role that the online schooling has played in increasing the equitable access to education, that we know, of course, is like one of the milestones for a person to grow in his personal path and also career. So intervention to reduce this gap will be fundamental. Thank you very much for listening to this presentation. I hope you enjoyed the presentation, and please feel free to write us any questions. If you have any comments, we are more than happy to answer. I leave the floor to Professor Folucci to conclude, and thank you again. Have a great rest of the day.
Yeah, it was a pleasure to be here with you all today, and unfortunately we can't be live because of the time difference. In a nutshell, we would like also to thank all the collaborators to the special issue from the Value in Health Economics and Policy group that brings together academics, experts, stakeholders from the globe. It's based here in the University of Newcastle, but we have a number of members at the University of Bologna, Italy, and other universities in Italy, and in fact in Brazil as well. You can follow our work on these websites and our social media, and write us if you have any question and interests about our work that goes beyond, but is very much committed to the collaboration between Brazil and Italy. We also have important developments in terms of helping countries with educational and training programs in health economics, management, and policy, which clearly is what was needed at the beginning of this pandemic. And certainly our authorities here and in Brazil, in Italy, and globally need to think very carefully on upskilling our administrative and hospital workforce to understand really how we can have integrated responses to pandemic of this kind, and even now more so that we are looking and witnessing the vaccine problems, I would say, dramatic in some cases in terms of access, and certainly in terms of mismatch between really the high demand and the rollout and the difficulties in distribution and also reproduction of them. So it is important we start understanding this. And for those interested in our programs, please connect to this link. I would like to thank you once again for your hospitality and looking forward to working together.
Thank you very much to all the speakers, and I shift to the Italian to present our last guest. E con grande piacere che lascio la parola al professor emerito Paolo Marchiorri Bus, direttore del Centro di Relazioni Internazionali della Fiocruz, al quale aspetta l'onore e l'onere.
Muito boa tarde a todos. É um prazer participar dessa reunião. Vou falar em português para a facilidade da minha comunicação. Eu gostaria de falar italiano, afinal eu sou Marchiorri, mas ainda estou aprendendo italiano para um futuro próximo, poder falar essa língua tão bonita que faz parte da minha história familiar, inclusive. Eu sou diretor da área de Relações Internacionais em Saúde da Fundação Oswaldo Cruz e quero aqui, em nome da presidente da Fundação Oswaldo Cruz, professora Anísia Trindade, saudar a todos os participantes desse evento, agradecer Fábio, agradecer o embaixador, agradecer também a Embaixada da Itália, o governo da Itália, por proporcionar essa reunião tão importante para a cooperação internacional entre Brasil e Itália. Eu gostaria de dizer que nós estamos fazendo um grande esforço na Fundação Oswaldo Cruz, como Instituto Nacional de Saúde Pública do Brasil, no enfrentamento da pandemia pela Covid-19. Nós, desde o princípio do processo, lá atrás, em janeiro de 2020, começamos a organizar esse grande esforço de enfrentar a pandemia aqui no Brasil e também na região da América Latina e com a África de língua portuguesa, espaços, territórios, espaços geopolíticos com os quais nós mantemos uma muito ativa cooperação internacional de desenvolvimento do conhecimento, a troca de experiências, e que tem sido muito útil, ainda que nós tenhamos que reconhecer a gravidade da situação da Covid-19 aqui no Brasil e também na nossa região. O Brasil, junto com os demais países da América Latina e os próprios Estados Unidos da América, lamentavelmente somos o epicentro da pandemia e temos, com uma população global relativamente pequena, o maior número proporcional de casos e de óbitos. Isso se deve, muito provavelmente, a questões estruturais, tanto da nossa situação epidemiológica, quanto dos sistemas de saúde da nossa região, e também por uma reduzida, eu diria, muito menor do que seria desejável, cooperação internacional na região da América do Sul. Em todo caso, temos feito um grande esforço e queremos, com a ciência italiana, com os pesquisadores italianos, com o governo da Itália, com a Embaixada da Itália, estabelecer vínculos e trabalho coletivo, comum, em conjunto, de uma maneira bastante profícua para que nós tenhamos ajudas no enfrentamento da pandemia que, de forma tão grave, está instalada no Brasil e na região. Resumidamente, a Fiocruz vem trabalhando desde o início da pandemia, em março, fevereiro de 2020, em 5 campos de trabalho centralmente. O primeiro deles é no apoio diagnóstico. Dado a grande capilaridade que a Fundação Oswaldo Cruz tem no Brasil, nós estamos em todas as regiões do país com nossos centros de pesquisa, com os nossos institutos, e também porque estabelecemos uma muito importante rede de laboratórios, já que a Fundação Oswaldo Cruz é a referência nacional para o diagnóstico, embora com o grande esforço que fizemos de capacitar pessoas em todo o Brasil, como órgão que somos do Ministério da Saúde. Hoje temos uma ampla rede de diagnóstico que tem a Fundação Oswaldo Cruz no epicentro dessa rede. Então, o apoio de diagnóstico se dá principalmente pelos laboratórios de vírus respiratório da Fundação, que é uma referência, inclusive, para a região das Américas, reconhecido pela OMS e pela Organização Pan-Americana, capacitando laboratórios, ofertando testes, plataformas de exames e também um apoio logístico à grande importância que tem a coleta e a resposta aos exames para o diagnóstico da Covid-19 no nosso país. Também temos um importante centro de atendimento hospitalar com 200 leitos, 200 camas, um centro aqui no Brasil, aqui no Rio de Janeiro, modelar, eu diria. Eles todos são leitos individuais, e nós já fizemos individuais e com separação absoluta entre os pacientes, com tratamento do ar dentro de cada uma desses boxes, que são um funcionamento de forma a garantir a biossegurança absoluta. Já internamos, já tivemos a oportunidade de receber mais de 2.500 portadores dessa enfermidade, com uma letalidade bastante razoável para o grau elevado de gravidade com que recebemos esses pacientes. Temos também trabalhado na capacitação das equipes de atenção clínica, tanto na atenção primária do Sistema Único de Saúde brasileiro, quanto no campo do tratamento intensivo. No campo da pesquisa e desenvolvimento, um grande número de pesquisadores da Fundação está envolvida em redes de vigilância genômica, em redes de diagnóstico, em redes de estudos clínicos. Fomos aqui no Brasil um dos polos do grande projeto Solidarity, o projeto que trabalhou opções diagnósticas, lamentavelmente, sem que esse projeto Solidarity da Organização Mundial da Saúde, certamente conhecido de todos, não conseguimos chegar a nenhuma terapêutica efetiva contra o vírus, mas conseguimos sim estabelecer protocolos clínicos que hoje estão sendo usados amplamente no território nacional brasileiro e também em cooperação com ministérios da saúde da região, difundimos esses protocolos. Na parte de comunicação, de informação e comunicação, é extremamente importante o projeto que temos do Observatório Fiocruz da Covid-19, acessível na sua página web, que além de fazer um acompanhamento diário dos casos e da distribuição dos casos no Brasil, também temos emitidos alertas que são esperados pela comunidade científica brasileira e também pelo mundo da gestão dos sistemas de saúde, pelas nossas conexões com o próprio Ministério da Saúde, evidentemente, do qual somos parte da estrutura, mas também com os conselhos nacional de secretários municipais de saúde e de secretários estaduais de saúde. Então, esse processo de difusão de informação, de protocolos, da situação atual, os melhores momentos, quais as melhores respostas aos diferentes momentos da pandemia, porque há que ressaltar isso, a pandemia no Brasil está constituída de várias pandemias. Por exemplo, o nosso centro Leônidas e Mariadine, o Fiocruz Amazônia, que está localizado em Manaus, foi vital na identificação da nova variante do vírus e de buscar soluções tanto para diagnosticar com o diagnóstico diferencial essa variante nova, quanto no rastreamento genômico na região amazônica das variantes que lá estão circulando. E, nesse sentido, especialmente, organizamos uma rede pan-amazônica de institutos nacionais de saúde capazes de fazer troca de amostras e fazer também aperfeiçoamento de pessoal e aperfeiçoamento da qualidade dos protocolos e na execução dos protocolos no diagnóstico clínico, sobretudo no sequenciamento genético do vírus, hoje tão importante para a vigilância geral da enfermidade no Brasil e, particularmente, para a vigilância genômica. Apoio às populações vulnerabilizadas é o quinto bloco, digamos, de ações da Fiocruz. Nós estamos localizados num lugar extremamente pobre do Rio de Janeiro e fizemos, naquelas populações de favelas que cercam a Fundação Oswaldo Cruz, na grande Rio de Janeiro, um conjunto importante de ações diretamente ligadas à atenção, ao suporte a essas populações vulnerabilizadas. E, finalmente, extremamente importante também, toda a questão da educação para a qualificação de profissionais no enfrentamento da pandemia. Nós temos um campus virtual muito importante, com um conjunto bastante expressivo de cursos à distância, curtos e médios, que permitem que centenas, milhares até, de profissionais estejam bebendo dessa fonte de formação pela educação à distância, já que estamos certamente prejudicados na educação presencial. Embora os nossos cursos de mestrado, doutorado, especialização continuem operando no modo virtual. Mas eu me refiro especificamente a cursos curtos para enfermagem, enfermagem de cuidados intensivos, vacinadores do Sistema Único na atenção primária. E agora acabamos de lançar um projeto muito importante, que é um mestrado e um doutorado em vigilância de fronteiras. Como sabem, o Brasil tem fronteiras secas, fronteiras terrestres, com grande permeabilidade da população, com pelo menos 9 outros países da América do Sul. Exceção de Chile e Equador, nós temos fronteiras com todos esses países e agora estamos muito preocupados diante, digamos, da manutenção num patamar muito elevado da pandemia, de capacitarmos profissionais para a vigilância de saúde nas fronteiras. Eu fico por aqui, penso que, resumidamente, dei a vocês um panorama do que a Fiocruz vem fazendo. Os meus colegas pesquisadores que me antecederam apresentaram alguns projetos muito importantes que estão em andamento da Fiocruz. Agradeço a eles por terem feito isso e quero colocar o Centro de Relações Internacionais de Saúde, a Fundação Oswaldo Cruz, não só à disposição para a cooperação, mas quero expressar-lhes o nosso grande, profundo interesse de expandir a cooperação com instituições italianas no enfrentamento da pandemia pela Covid-19. Muito obrigado, devolvo a palavra ao coordenador dessa sessão.
Ringrazio il professor Busse per le sue parole e mi permetto di dire proprio un pensiero mio. La battaglia contro il Covid si sta combattendo hic et nunc, come ti dicevano i latini, qui e adesso in Brasile, e la scienza italiana c'è. E spero che ci sia ancora di più nelle prossime settimane e nei prossimi mesi. Vi ringrazio a tutti, ringrazio tutti gli intervenuti, le persone che ci hanno seguito sulle piattaforme, ringrazio Simona, Priscilla e Luisa per l'aiuto e veramente grazie a tutti i colleghi per quello che stanno facendo.